Hello, good afternoon, everybody.
Hello, good afternoon.
Good afternoon.
Hi, all together.
Hello.
Great to see you.
Yeah, we didn't see you yesterday, but happy to see you today. That's the most important.
Yeah, I thought it's optional, and I've
Yeah,
been
sure.
at a PhD defense yesterday, so I think the Zoom thing will work with me.
Yeah, okay.
Hello, everyone. Thank you
Hi.
for coming. I just want to let
Hi.
you know that also some of the participants are already connected. So we will wait 10 minutes and around 10 minutes more, and then we should start. So I put already this kind of background instructions for the participants. So dear participants, thanks again for your participation. I kindly invite you to have a look to these instructions I'm displaying, and we will start in about 10 minutes. So we still have 10 minutes again to relax before before the party starts. Thanks again to all panelists for joining. Indeed, nice to meet you, Axel. Now we meet in 10 minutes or so. Thanks again, host.
No problem. Just a reminder, Axel, this morning, normally you're the second speaker. Hanan should have been the first, but unfortunately, but maybe you saw it also, she's ill, so we will directly start with your speech. So, but anyhow, I will introduce you. So, but just to let you know that you don't have to wait, but I will introduce. So no problem.
Okay, great. So do I have more time than?
No. Up till this moment, my answer is no.
Okay, great.
No, we'll try to stick to the time. I hope it's everywhere nice weather, so maybe we'll try to finish it a bit earlier, or we see what we can do with the Q&A and so on. So, but if you need 1 or 2 minutes more, it's no problem.
Okay, great. Thanks.
Hello to all. Excuse me.
Hello.
Good
Hello
afternoon,
to you as well.
Maria.
Hello.
Maria. Yeah.
Hello, Maya.
Hello. Hello. Thank you. So I'm also ready. So whenever you start, fine
Yeah,
with me.
I think we will give like 10 minutes or yeah, 6 minutes or so. We will start.
Okay. I
Just give
have only some this welcome speech, like 3 minutes, not more, not more.
Good. Thanks again for joining.
Thank
Okay.
you.
Bonjour.
Bonjour. Bon appétit.
Merci. We had the PhD defense this morning, so I just finished the PhD defenses.
Okay, but you still have time. You're just the last one, so you can.
Yes.
You still have time to keep yourself together, so no problem.
Okay.
Do we open up the line, Filippo? It's 1 o'clock.
Hello, I just 1 more minute. I see the participants are increasing. Just let's allow 1 more minute and we will start. Okay. I will
Okay.
give you a sign shortly. Good. Well, I think we should start. We have about 150, even more participants already connected. I think more will join shortly.
Okay.
Of course, I will give you the floor. I will stop sharing now.
Yeah. Ladies and gentlemen, good afternoon and welcome to this webinar on the determination of the clay content of soils. My name is Joost Salumi and I'm working as a soil protection expert at the Department of Environment and Spatial Development of the Government of Flanders. And I will be your host for this webinar. This webinar will be recorded, so by attending this webinar, you consent with this and some more practicalities will be given at the beginning of the webinar. As you have seen on the agenda, we have a tight schedule with a lot of interesting speakers. So we start right now with a short introduction by respectively Filippo Benedetti, coordinator of the Global Soil Laboratory Network, BESOLAN, Maria Romic, chair of the European and Eurasian Soil Laboratory Network, BESOLAN, and Christophe Thierry of the Belgium and Luxemburg Soil Laboratory Network, BESOLAN. The three of you have 10 minutes all together. So please now, Filippo, Maria, and Christophe, the floor is yours.
Many thanks, host. I would like to extend again my greetings to all participants for your participation and to all panelists for their availability to participate and share their knowledge. I just want to share a very quick presentation I prepared for today. So I hope this visible is charging. Just a second. Yeah, there you go. Should be able to see that. Yeah. Again, my name is Filippo Benedetti. I'm working at the Global Soil Partnership Secretariat at the Land and Water Division at FAO here in Rome. I'm coordinating this global soil laboratory network called BESOLAN. I think most of you are already familiar with that, as I guess most of the participants today are part of the network. But I would like to give a little bit of background just to give as need the framework in which this webinar is organized today. Basically, the global soil partnership is a partnership. And it is a mechanism established in 2012, hosted by FAO. Its main mission is to promote sustainable soil management and position soils in the global agenda. Because soil are gaining interest, they are very important for many SDGs, for many activities. It's a cross-cutting topic, and in order to get reliable data to base sustainable soil management practices, to formulate recommendations to farmers, we need indeed good laboratories. Laboratories able to provide accurate, precise results. This is why BESOLAN was established in 2017. The goal is to group together laboratories to improve the analytical capacities. And this has been done over the last 6 years, even more now. And we have about 1,100 soil laboratories registering the network from 163 countries. You can send them up how many laboratories where they are distributed. Basically, the network is getting very big. So we are organizing the network into regional networks. So there is a regional network for Europe, one for Asia, one for Africa, and so on. But also at national level. So we are grouping laboratories into working in the same countries. And this is the case of today. This webinar is organized by the laboratories operating in Belgium and Luxembourg. So they're grouping together into what is called Belgium and Luxembourg Soil Laboratory Network. And today, basically, we are trying to they are trying to sharing their experience on and their knowledge on clay determination. So this is an example, basically, on the kind of activities that can be organized within the national networks, but also shared between within the region. So, for instance, these activities falls within the activities of the European and Eurasian Soil Laboratory Network, but of course, we invited all laboratories from BESOLAN to participate. So please feel free to register in the network. This is just an overview of the activities we're organizing. So of course, everything related to the measurement of soil parameters. So there is all the activities related to the harmonization of protocols, capacity building, also about health and safety, not only on procedural details, quality control, which is key, of course. Glossolain organized many proficiency tests and encourage countries and regions to do that. But of course, also we train laboratories on internal quality control procedures. There is a core activity on equipment and especially on soil spectroscopy called Glossolain Spec. And of course, now we are starting also dealing with laboratory management, data information system and information system, so the so-called LIMS. So now I will give the floor back to host. I just want to stress again there's some rules of today. So there is the chat, you can write your messages there. There is also the question and answer section. There you are able to type your questions and panelists will be able to answer that. I already have to say that unfortunately we cannot release any certification of attendance for this kind of webinar, but the recording and the presentations will be available in the following days on the BESOLAN webpage. By doing that, I wish you a fruitful webinar. We will meet again in the end for the conclusions. I just want to tell you that this is one of the webinars that Glossolain is organizing. There will be one in the first week of July on CC, organized by the African Soil Laboratory Network. You will be welcome to attend as well. We will share some information soon. But again, the network is active, the members are active, so this is really the idea behind the network. So laboratories grouping together to join activities and sharing knowledge. So thanks again for your participation. Thanks to the panelists, and back to you, host.
Yeah. So Maria, can you continue, please?
Oh, hello. Good afternoon, everyone, dear organizer, presenters, participants. My name is Maria Romic. I'm a soil scientist from the University of Zagreb, Croatia. And it's my great pleasure to welcome you all to this important webinar organized by the Belgian and Luxembourg Soil Laboratory Network, BESOLAN, on the topic of clay determination. As the chair of BESOLAN, I'm delighted to such a strong turnout of expert researchers and laboratory staff from across our soil science community, let's say. Determining clay content is an essential for understanding soil properties, which in turn plays a crucial role in managing soils for both agricultural and environmental purposes. Laboratory analysis of soil texture, including the determination of clay content, can be challenging due to the complexity of soil composition and potential interferences. And however, advancements in analytical techniques and technology have significantly enhanced the accuracy and reliability of this analysis. So to achieve precise and meaningful results, it is vital to employ consistent methodology, ensure proper sampling handling, and remain aware of any potential sources of interference. Today's webinar demonstrates the shared commitment of global Soil Laboratory Network of Glossolain and the European Soil Laboratory Network BESOLAN to supporting laboratories and improving their performance, fostering collaboration and knowledge exchange, which is an integral aspect of the network mission. This cooperative approach aims to enhance the overall quality and consistency of soil analysis, thereby contributing to better soil management practices globally. I would like to extend my sincere gratitude to the BESOLAN members from Belgium and Luxembourg in getting together in this National Laboratory Network BESOLAN for organizing this event and bringing together such a distinguished group of speakers and participants. So your efforts are invaluable in fostering collaboration and knowledge sharing within the community for the benefit of all. Let's hope other countries could do the same. As we proceed with today's presentations and discussions, I encourage all of you to actively participate, ask questions, and share your insights. And once again, welcome to the webinar. I look forward to fruitful engaging sessions. Thank you.
Thank you, Maria. So Christophe, can you please take the floor?
Yes, I will. I will also share my screen. I just have presented 2 slides, if everything goes well. Okay. So also from my side, a very warm welcome to this webinar. We're coming down from the global to the interregional and then finally to the national level. So just a few words about BESOLAN. So
sorry, Christoph, I think we have issue with your screen because we see your email.
Sorry.
Yes.
Yeah.
Stop sharing. Sorry for that. I'll start again. Share. Can you tell me what you see now?
Yeah, it's loading.
No.
No, we see very zoomed slideshow. Yeah, now it's okay. Can you put on presentation? Yeah, presentation mode.
Use presentation mode, and now it's fine.
It's loading.
No.
I think it's.
No.
Can you try to click? Maybe you're in the last slide. I don't know.
Maybe you see something now. It's better.
Yeah, perfect. Thanks.
Okay, sorry for that. Okay, so once again, just some words about the BESOLAN. So basically, we are the Belgium and Luxembourg Soil Laboratory Network. We were established more or less 3 years ago. And basically, the main idea of the network has been said by Filippo and Maria as well, is to facilitate exchange of information, and then especially between soil experts in Belgium and Luxembourg. So our motto is more or less, think globally, but act locally. All of you are invited to register freely. You can go to our website, BESOLAN, under the FAO, where you will find the links how to proceed. So the BESOLAN, we consist, or the coordination is done by the 4 reference laboratories of the different regions in Belgium. So on the one hand, you have for the Walloon region, the Liège Université Jean Blou. For Brussels, you have the Brussels Environment Administration. From the Flemish side, you have VITO. And in Luxembourg, you have le gouvernement du Grand-Duché de Luxembourg, ministère d'Agriculture, de la Viticulture et du Développement Rural. So these are the 4 reference libraries, and we really hope we have set up this webinar this afternoon with very interesting speakers. So we hope you will all enjoy it, and please come back with questions or others later on if you have any. Last but not least, I would like to thank our sponsors, which are the regional and national governments we are listed here under. And now I would like to give back the floor to you, Joost, so that we can start with the webinar. Thank you.
Thank you, Christoph. Filippo, you want to say some words in between? Some practicalities? Or is everything running smooth?
Well, I think already said more or less how to use the question and answer and the chat. We just want to encourage again participants to use the questions and answer chat box to interact lively with the panelists. Your answer could be answered there or eventually picked up to have to be discussed in the open session later on. But please really feel free to encourage there to interact there.
Okay, thank you very much. So after this first introduction, knowing now who we are, BESOLAN, Eurosolan and GLOSOLAN, we will start with our first topic, and this first topic deals with the importance of the clay content of soils. Of course, it is important. Our first speaker should have been Hanan Arui Bugbida, both the vice chair of GLOSOLAN and a soil physicist. So having a broad view on the topic. Unfortunately, this morning I was informed that she's ill and will be unable to speak, of course. Wishing her a quick recovery, I can announce, as already said, that you shall make her presentation available, so you'll get at least all the information. So I'm very sorry, but this was only happening this morning, and of course, no other speaker could have been found at this short time of period. So that brings us directly to our next, but now first speaker, and that's Axel Don of the Thünen Institute of Climate Smart Agriculture in Germany. He's the head of the research unit Soil Organic Matter, and why have we chosen him? He will make the link between the topic of the webinar of last year, being carbon, and the topic of this year, being clay. It's always good to have a bridge between the different webinars. But as most of you will know, of course, one of the indicators of the proposed soil monitoring law deals with both aspects. And we like to hear Axel's opinion on this or another soil health indicator, but of course, based on soil organic matter and clay. So please, Axel, you have the floor for about 20 minutes.
Yeah, thank you, Jos, for the friendly introduction. And thank you for inviting me here to talk about carbon and clay, and carbon and clay as indicator for soil health. So I hope you can see my screen now. I want to bring you first from the lab to the field with two soil profiles. One is a sandy soil on the left side, a cambisol, and one is a Chernozem, which is a silty or clay-rich soil. And you see different soil properties. And here also the carbon content is here. On the left side, the cambisol has 1.2% carbon. The more fine textured soil, 2% carbon. They are both used for agriculture, and therefore the plant available water is really important. And you see the huge difference between these two soils. The left side cambisol has only roughly 80 millimeters plant available water in the first meter, and the Chernozem more than 200. And it's all related to texture and also a little bit to carbon. And we need to be aware all the time about the diversity of soils we are dealing with. Clay content is a central soil property, that's for sure, and it's hardly affected by land use and climate, and therefore it's different from carbon. Clay is a result of the pedogenesis and the bedrock, and it's pivotal for the water retention function and for the stabilization of carbon. And to the right side, you see that at the really nano scale, it's the clay organic matter complexes that stabilizes the carbon. And also at larger scales, it's the aggregates that are really a result of soil texture and carbon content, and important to stabilize carbon and to really help to maintain carbon in the soil, or to even build it up and to help with carbon sequestration. At a larger scale, if you look at European scale, clay is really the key parameter to predict soil carbon. And this is done in a new study here with Lucas data at European scale. And the clay fraction really is the most important variable to predict carbon content. And on the right side, you see a relation between the soil organic carbon content and the clay content. And you see there is a relation. And I would rather say the clay content really shows us the lower limit for carbon. So the higher the carbon content, the less likelihood is it that you have really low carbon soils. And we investigated this also at our national inventory in Germany, where we sampled more than 3,000 agricultural sites, so grassland and cropland and permanent crops, down to 1 meter depth with more than 100,000 samples. And it was finished in 2018, and the data are freely available, open access available. And we're just about to resample all these sites. And results are really surprising here, first with the carbon content or the carbon stocks in the topsoil and in the subsoil. And you see the high scatter and variability. So if you go from one point to the next point, which is only 8 kilometers apart, you can hardly predict what carbon content you find at the next point. So we have a huge variability and only a few regional patterns. And in the subsoil, it's even it's even worse, and you cannot really predict what you find in the next soil pit. It's totally different to soil texture. And these are the same sampling points, but here you see very clear regional pattern. You see in the north of Germany, we have all the glacial deposits, which are sandy. Then we have a silt band here in the middle, and in the hilly regions of Germany, we have a mixture depending on the bedrock. And this is produced or generated into such a map for the clay content. So clay is less variable than soil organic carbon. Maybe it's a good message for all of you focusing on the clay content and measuring clay content. And we need to be aware that we do not cover all possible texture classes similarly, particularly in agriculture. In Germany, we have really a strong focus on the sandy soils and the silty soils, and we rarely have soils with more than 45% of clay here in the violet area. So we have no even distribution. And I can tell you from our inventory that soil texture analysis was the most time-consuming analysis among our standard soil parameters. So it took us 7 years for the whole inventory. And when the whole inventory was finished, the texture analysis was still going on because it was so time-consuming. Therefore, investing in good analytics and also faster analytics is really worth. And of course, we had an automatic system, but still it was quite a workload. Results are interesting. Here for Germany, we also see the relationship between the soil organic carbon stocks and the clay content in the black dots here. So the more clay we found, the more carbon we found. But we also have these red dots here. And these are very sandy soils with less than 10% of clay and very high carbon content, at least in some of these soils. And they are all located here in the north of Germany, close to the border to Denmark and to the Netherlands. There you can find similar soils, and we call them black sands, sandy soils that have really a high carbon content. And we can distinguish them with also a very wide CN ratio, wider than 13. And we digged into the land use history of these sites, and we've seen that most of them has been heathlands or peatlands some decades or even centuries ago. So the heathland history, the heathland litter generated such high carbon content that is still abandoned in these soils. Today, they are all croplands and grasslands, and you cannot see any heathland or peatland anymore, but you see it in the soil with this high carbon content, and that something strange was happening in these soils. Here, and it's not only the carbon content that is closely related to the clay content, but also the carbon quality. And here we determine the carbon fractions, so the particulate organic carbon fraction, or we can also call it light fraction. And we see the relation here: the higher the clay content, the lower is the particulate organic carbon fraction, meaning the remaining carbon is all mineral associated, so the mineral associated fraction. And we also see this regional pattern that here in the sandy soils in the north, we have all these particulate organic carbon. And if we want to predict the carbon quality, so the fractions, we again need the texture, the clay content and the sand content, and then we can very nicely predict the fraction of particulate organic carbon in the soil. Going back to our two soils, the question is, are these soils in healthy conditions or are they degraded? And that's a tricky discussion. And there is a nice report from the EEA for European, from the European perspective on indicators and thresholds on soil health. And it's for sure, soil health indicators should be sensitive to land management and climate. That's what we are talking about. We want to manage our soil sustainable and want to know what happens due to climate change to our soils. And we need indicators to tell us something about our land use and climate. They need to be relevant to soil functions, because that's also what we want. We want that the soil functions are in good shape and that they are working, and therefore indicators should have a connection to the soil function. And of course, they should be measurable, they should be accessible, and in the end inform land managers and policymakers. And the carbon to clay ratio has been discussed widely as an indicator, and Joost mentioned it, that it's already in the proposed European soil monitoring law as an indicator for soil health. It's really an attempt to normalize the carbon content for the impact of the clay content. So to really get rid of the influence of clay, let's say like this. And the good thing is, it's also related to physical parameters, like here the core West score, which is a parameter judging on the quality of the soil structure, showing that the higher the C to clay ratio is, the higher is the core West score. We used this carbon to clay ratio and applied it to our German data set, which is almost 3,000 mineral soils. And we were really surprised, because if you look here, the clay soils up here were mostly classified as degraded, almost all of them. And the very sandy soils down here are almost all classified as very good or good. And if you see this, you already see there's a bias in there. It cannot be that all farmers working on clay soils did a bad job and all farmers working on sandy soils did a good job. And we had to conclude that the normalization with the clay content is too strong. It's not really a fair judgment and a fair normalization. And therefore we propose something different. And this is the measured carbon content divided to an expected carbon content or a typical carbon content, and this typical carbon content is very closely related to clay and can be also derived by a single regression between carbon and clay. And with this new alternative ratio, we see that we got rid of the bias. We see that in sandy and clay soils, we both has degraded and non-degraded soils. And you can read more into this in our paper published in Soil User Management. And even if we look at the effect of this indicator on the soil structure, this is the carbon to clay ratio, so the proposed one, and the relation to the bulk volume, so the pore volume. And this is our new alternative ratio, the carbon to carbon expected ratio. And our relation is even stronger, showing that our indicator is even better as an indicator for a good soil structure that is important, for example, for erosion control. Therefore, we had to conclude that the carbon to clay ratio should not be used as a simple soil health indicator, but you should be really careful because it has some bias in there. Yeah, last but not least, I want to show you that there's also another option to analyze the soil texture. And we were amazed how good our soil scientists were to determine the texture analysis with their fingers. You know, finger texture by feel. If you're in the field and if you're a well-trained soil scientist, you can really find out how much clay is in the soil by rolling the soil and by really feeling it. And some good soil scientists, they also take it between their teeth and feel how much sand is in there. And we were surprised that the clay fraction here for the different soils was very well analyzed also by texture by feel. We need measurements if we need a high precision, for sure. We need the lab. And it's good that we talk about lab measurements today. But for some analysis, it might be enough to have a rough estimate. And this can be done in the field. And therefore, we need good soil scientists who are able to determine clay content also with texture by feel. And it's also published if you want to read more about this. I want to conclude, so clay is really a key soil parameter and a key parameter for the soil quality, and it's closely linked to the soil carbon and also the carbon quality. So the particulate organic carbon is related to the soil texture. And you've seen, I gave out a warning that the carbon to clay ratio seems to be a biased indicator, and we should really further develop it to develop something that can be applied to all soils to a broader context at continental or even global scale. And the clay content need to be accounted to differentiate between the manageable, which is the soil health, and the static, which is the soil quality, soil parameters. So we cannot change the plant available water. That's really related to the clay content and the texture, and that is soil quality. And carbon is more related to soil health because it's manageable, we can change it. And I hope I was able to show you that both the clay and the carbon are closely linked together. Thank you for your attention.
Thank you very much, Axel, to give us this very nice talk about as well the importance of clay and soil organic carbon. You've given us a nice overview also about soil health indicators. Thank you very much. I just want to remind everybody that you still can put your questions in the Q&A, and we will answer them after when we have our first Q&A session. So thank you once more. And so we can go on to our second topic of this afternoon, and that's about the standard methodology for the determination of the clay content of soils. Of course, an important part of this webinar is to give you an insight into how the standard methodology of clay determination is performed in our laboratories. And different colleagues of Belgium and Luxembourg have put all their expertise together. We have worked quite a bit on that. And Chris van Hove of the Flemish Reference Laboratory for Environmental Monitoring, VITO, will guide us through the whole procedure. Chris is a senior researcher, and her work focuses on the development, validation, and publication of inorganic analytical methods. Her talk is in cooperation with Lionel Ledet of the Luxembourg Ministry of Agriculture, Food and Viticulture, and Gilles Collinet of the Université de Liège of Wallonia. Chris, because of this topic being very important, you have the floor for 40 minutes.
Yeah, thank you. I hope you all see my presentation by now. I will present you the standard methodology that is applied by the reference lab in the Belgian regions and also in Luxembourg for the determination of the clay content of soil. In this presentation, we mainly focus on the analytical aspects of the clay determination. If you have a look to the background, the Belgian soil map was drawn up based on the results of intensive soil mapping that was done during the 1950s and the 1970s. And at that time, the soil profile was studied down to a depth of about 1.25 meters. Also at that time, a lot of attention was paid to the optimization and the development of the analytical method. If you see at the right side, the picture of a pipette is already drawn over there. And at the bottom, you see an example of a soil map, part of Belgium that is shown, and they are available online if you would have a closer look to that. For the soil classification, the soil triangle is used also, and it's based on the relative proportions of sand, silt, and clay, whereby the sand fraction is the fraction between 50 micrometer and 2 millimeter. The silt fraction is the fraction between 2 micrometer and 50 micrometer, and the clay is the fraction less than 2 micrometer. It has to be said that depending on the country, sometimes also the 63 micrometer instead of the 50 micrometer is used as the limit to separate the silt fraction from the sand fraction. Also in Belgium, and especially in the Flanders part, the percentage of clay is not only used for the soil classification, but is also used for the recalculation of soil remediation values for some heavy metals. So it's a different purpose than for the soil classification. Which method is now used in our reference labs in the different parts of Belgium and Luxembourg? It's all done by the same method. It's the determination according to the ISO 11277 method. And it describes the determination of the particle size distribution in mineral soil material, and it's the method by sieving and sedimentation. I think it's a well-known method by everyone. In this presentation, I will guide you through the different steps of the procedure, highlighting some critical points and also showing the variability within the procedure. What is the principle of the method? The analysis is done on a soil sample, dried at 40 degrees C and grounded less than 2 millimeter. After removal of the organic fraction, the carbonates and the soluble salts, the mineral fraction is left over, and it's on that mineral fraction that we do the separation into the different particle classes, and that by wet sieving and sedimentation followed by the pipetting. Prior to this step, a peptizing agent is added, and it's done to disperse the particles as much as possible. After that, a separation is done of the sand fraction by wet sieving through a sieve of 50 micrometer. The suspension is then transferred into a sedimentation column to separate the clay and silt fraction by sedimentation, and that according to Stokes' law. What does this mean? That is that the larger a particle is, the faster it moves in water under the action of the gravity. This is done by a constant temperature, and the pipetting is also done twice at a fixed depth, and we take it always a depth of 10 centimeter. The first pipetted volume contains the clay and silt fraction, while the second one only contains the clay fraction. If we have now a closer look to the procedure, these are the different steps that I will guide you through. First of all, the sample preparation, how we do the destruction of organic matter. Also, the removal of carbonates and soluble salts are demonstrated, and the dispersion step is discussed. And then it's following of the wet sieving at 50 micrometers to separate the sand fraction. And the sedimentation is then highlighted for the pipetting of the silt and the clay fraction. And then, of course, the calculation of the results. What about the sample preparation? The soil sample is dried at 40 degrees sieve. Then it is gently stamped so that the clumps are removed, but you always have to be careful that you don't disturb the particle size. Afterwards, it's sieved through a sieve of 2 millimeter, and it's on that fraction, the fraction less than 2 millimeter, that the clay content is determined. If you look to the intake of the dried sample, according to the ISO method, 30 grams of sandy soil and 10 grams of clay soil is described, but we see that in the reference lab, the intake varies between 10 and 20 grams, and it's different than what is prescribed in the ISO. Also, we see that for clay salts, we sometimes reduce the intake even to 7.5 grams. Why this is done? This is done to get a better dispersion. We know that with a high content of intake, then the dispersion is not properly done with the clay sample. You also see that, for instance, we can work with a plastic bottle, and this plastic bottle can be used through the whole pre-treatment step, even for centrifugation, so you don't have to transfer your sample in between. The first step in the process is also the destruction of the organic matter. There are different methods available in the ISO standard to perform this. You always add hydrogen peroxide. And in this first method, we describe what is already described in the beginning of the ISO standard. And there 100 milliliter of hydrogen peroxide is added in steps of 25 milliliter. And it's always done with small steps to avoid the formation of too much foam, as shown in the picture on top of it. Afterwards, you cover the recipient with watch glass to let it stand overnight at room temperature to get already a first reaction. Later on, it's increased to a temperature of 70 degrees C, as shown here, using a hot plate. And then it is maintained during 50 to 20 minutes at that temperature to remove the organic matter. You have to take care that the volume of this suspension should always be kept at around 150 milliliter, and that is done by adding demineralized water so that you have your process under control. Another method that is recently included with the last revision of the ISO standard is the use of the ultrasonic bath. Why it is used? It is used also to speed up the process of the sample pre-treatment. And therefore, we also add 75 milliliter of hydrogen peroxide in small portion. These additions are spread over 3 hours so that you have added slowly and the direction is not too vigorously. And every time the addition of hydrogen peroxide is done, you do a 1 minute ultrasonic power at a temperature of 70 degrees C. The use of ultrasonic bath is included in the last revision of the ISO, and it's added to the annex. It has to be noted that you have to compare also the comparability with the original method, and that is done because in the reference soil database, it is important that you always have comparable data. And we have shown that the soils in our region, that we have similar results with both methods. We apply these methods already for several years, and we have obtained comparable data. The next step in the process is the removal of the carbonates. Also here, different methods are available in the ISO standard. The first one is the use of a hot plate. At the beginning, 10 milliliter of hydrogen chloride is added, 1 molar of hydrogen chloride to destroy the carbonates. For 1 hour, the reaction takes place at room temperature, and then there is a gradually increase of the temperature up to 70 degrees C to destroy all the carbonates. Also, the solution is boiled for at least 10 minutes, and then it is cooled down. Also, for the removal of the carbonates, we introduced the ultrasonic bath power and used the ultrasonic power to destroy it. We also do in advance a check of the original sample to check the presence of the carbonates. So we add a little bit of hydrogen chloride, and if there is no sparking reaction, we can say that there are no carbonates present, and we just add a basic amount of hydrogen chloride. If there is a reaction when you add the hydrogen chloride, we know that carbonates are present, and then we add the hydrogen chloride. We use an ultrasonic power for 1 minute at 70 degrees C, and we leave it then at the heated bath at 70 degrees C for a while. After 10 minutes, we check the pH with the pH strip. And if it's below 3, we can say that the removal of the carbonates was successful, that all the carbonates were gone. If the pH is still above 3, we have to add additional hydrogen chloride to remove all the carbonates. Nevertheless, the reaction is divided over a period of 2 hours. So after 2 hours, the carbonates are removed in this procedure. Once the carbonates are removed, we can then remove the soluble salts that are available. So we add some water to the recipient to remove and to wash the suspension. and then it's centrifuged and the supernatant is removed then. We always check the electrical conductivity of the supernatant, and it has to be less than 40 millisiemens per meter. If it is above, we repeat the step, and this step is repeated until we have the low electrical conductivity. This is important because afterwards you do the dispersion, and for that you need a clear supernatant. So the next step we apply is the dispersion. On the sample, 25 milliliter of dispersion aided is added, and also some water is added to the sample, and it's shaked thoroughly. Also here we use ultrasonic power in the water bed for 10 minutes at 70 degrees C, as shown in this picture. In the ISO standard, also other methods are available. It's in the original one, it was defined that you can do the dispersion at room temperature by shaking, but then it has to stand overnight. So it takes a long time. That's why we also introduced this ultrasonic power to speed up the pretreatment step. And it's also possible to boil the solution with a dispersion agent for 5 minutes. That are also possibilities to do the dispersion. Once the dispersion has been completed, then we can start with the separation of the sand fraction. It includes a wet sieving at 50 micrometer. So the dispersed suspension is transferred from the centrifuge bottle quantitatively onto the sieve, and then it's washed with water to remove everything. Also gently rub with the fingertips to not to push it through the sieves, but to give the possibility to get through the sieves. So it has to be washed sufficiently so that you become clear water. It's important that the total volume of the washing may not exceed 1,000 milliliter, because it's that sample that we have to transfer further to the sedimentation columns. The sand fraction is afterwards quantitatively transferred to a weighing vessel, as shown in the picture above. And it's done by using a gentle spray of the wash bottle. And then it's dried at 105 degrees C until a constant weight. And that is normally done by drying overnight. The next step is the separation of the silt and the clay fraction. For that, the suspension is transferred to the sediment cylinder, as shown at the right side. The total volume of the sediment cylinder is filled up to 1,000 milliliter exactly. And also in case that you use a magnetic styrer, you insert the magnetic styrer. The sedimentation cylinder has to be placed on a constant temperature environment. You can use to work with a waterbed, for instance, at 30 degrees C, as shown on the right, or you can work in a room with climatization. If you work in a room with climatization, then of course you can go immediately through to the pipetting procedure. Otherwise, you have to wait a moment and overnight, for instance, to have a constant temperature bath, and also that your sample is at a constant temperature. Important to notice is also that we include a blank cylinder, and the blank cylinder only contains water with a dispersion agent, and that value also we need for later on to correct for the calculations. So once the sedimentation cylinder is at a proper temperature, you can start the separation of the silt and clay fraction by pipetting. You place the cylinder or on a styrer, or you can shake the cylinder also up and down vigorously so that the solution is in a good suspension weight. And then you can stop the stirring, and then you put the pipette into the cylinder, and it has to be 10 centimeter below the upper part of the suspension. Note that the pipette has sideways opening so that when taking the sample, you don't disturb the suspension. Once the pipette is inserted, you take 20 or 25 milliliters, it's depending from the laboratory, is aspirated using the Robinson-Cohen pipette. Also note that this volume has to be calibrated so that you can work later on your calculations and perform it with the calibrated volume. The volume is then transferred to a weighing vessel, as shown over here. And this vessel is then dried at 105 degrees C until a constant weight is reached. So in this vessel, you contain the silt and the clay fraction. Then the second sampling is the clay fraction that you determine, and it contains also the dispersion agent. As I told you before, the sedimentation is based on Stokes' law, and that means if you use a constant temperature, you have a certain insertion depth, in this case 10 centimeters. You can calculate the second sampling, and in this case, if you work at 30 degrees C, this sampling has to be done exactly at 6 hours and 9 minutes after the first one. So you have to enter the pipette into the solution, into the suspension, 30 seconds before the end of the sedimentation time, and then you can do the insertion at the depth of 10 centimeters, and then you can aspirate 20 or 25 milliliter using the pipette. Also here, the content of the pipette is empty in the weighing vessel that is subsequently dried at 105 degrees C until a constant weight is reached. As you can see, these are all manual steps, and that takes quite some time to do this. It's also labor intensive, so you have to do a lot of manual pre-treatment steps. Therefore, there is also some room for automation and to work with an automated robot. Maybe first to say a part of the calculation, it's also important to know that the fraction of clay is determined on the mineral fraction, and this after removal of the organic fraction, the carbonates, and the soluble salts. This means that the total mass that you use, it's the sum of the sand fraction, the silt fraction, and the clay fraction, and that differs on the initial fraction because the 3 parts of organic fraction, carbonates, and soluble salts are removed. So by this, you can calculate your percentage of clay. As I told you before, the introduction of automated procedure is an advantage so that you don't have to be always working on the different manual steps. I will show a picture, a movie that I have received from Labo de Vlieger and Van Voren. It does not contain all the steps and not at the regular speeds, but is for especially to give you an idea of what steps can be automized and how that the procedure can be done. In general, this procedure takes about, in practice, 24 hours, and it can all be programmed with the computer, with the software of the computer. So there is a possibility first to wash the sample, but also to add the hydrogen peroxides, as you see by this automation. In the next step, the sample is heated, and you get the destruction of the organic matter. There is a rinsing of water. In between, water can always be added so that you can avoid an intensive reaction. Then hydrogen chloride is added to remove the carbonates. And also here, the reaction can take place. Afterwards, water is added for the washing procedure and for decanting. So once you have enough water over there, the water is removed. And while removing the water, the conductivity is monitored. You can, when the conductivity is not below 40 millisiemens per meter, the washing procedure is repeated, and it is repeated until the conductivity is below the prescribed value. Afterwards, and what's not shown in the picture, in the movie, there is also the possibility to automatically add the peptizing agent and to boil it also so that the sample, the suspension, is ready for the separation of the sand fraction. So you can take this sample for doing the sand fraction. If you go further for the pipetting, there's also a robot possible. This is the pipetting robot. Also here you place your cylinder and you can add there the suspension after you did the separation of the sand fraction. You add water until 1 liter, and you have also the possibility to stir the suspension very good. The styre goes up and down so that the homogenization is done through the whole cylinder and that you can see it then. Once the homogenization is done, the styre stops and then the pipetting can take place. It will be shown in a moment. Now the homogenization stop. The styre is removed from the solution, and then we go over to the pipetting robot. And as you see, here is the pipetting, and you can pipette 20 milliliter from the cylinder. And in front, you can see the weighing vessels in which the solution is pipetted. Afterwards, you can evaporate on these places already a part of the water content, but later on the vessels are transferred to an oven to do drying at 105 degrees C overnight to be sure that you have a dried sample. So now the robot is moving to the weighing vessels, and so that you can do the pipetting of the 20 milliliter. Now we have seen the different steps in the procedure, the pre-treatment steps, the pipetting itself. What about the reproducibility results that we obtained? First of all, we take a look to the intra-laboratory reproducibility results. So that is the deviation within the lab. And this is a typical example of a control chart for the full process of the clay determination according to the ISO method, and this applying the manual pre-treatment steps. 30 points are selected, and for that we obtain a relative standard deviation of 9.3%, and it's nicely distributed over the mean value. We have also give an example of the reproducibility results for the automated procedure. And for this part, the percentage of clay is calculated related to the initial intake so that you really see what is the relative standard deviation of the automated pre-treatment steps. And then for 30 points, you obtain a relative standard deviation of 3.8%. So you can see that with automation, you can also have a very low RSD value. We also organize round-robin trials, and these are organized on a yearly basis by FITO, and it's commissioned by our Federal Waste Agency, OVAM. In total, about 9 labs participate to these round-robin trials, and that are labs coming from Belgium, the Netherlands, Germany, and they all apply the ISO method, but of course with different pre-treatment steps. And it's also a combination of manual and automated methods. If you see to the results, these are results obtained since 2012 up to now. And the green bars refer to a reference sample. It means that the lab received a sample that is already homogenized, already sieved less than 2 millimeters so that they can directly start with the determination of the clay content. While the blue bars represent real samples, and for these samples, the lab itself has to do the preparation starting from the beginning. So there is an additional step included. The bars represented the clay content showing at the left side, and the orange line represents the RSD value, and it's showing on the right side. And as you see, the RSD values fluctuates a lot. It's depending also of the concentration, but it also depending on the type of the clay sample, of the sample itself. So it varies between about 8%, even up to 40%. So there can be a large variation in. What is the reason of this variation? It's very difficult to allocate. Of course, the different pre-treatment step can play a role, but also the type of soil sample. If we have looked to a summary to all these, what we have discussed now, then we see that the ISO 11277 is the reference method that is applied by the Belgian regions and also Luxembourg. The method is applicable for the soil classification, but in Flanders also applicable for adapting soil remediation limit values. And if you want high throughput samples, analysis samples, and especially in the framework of sanitation, it's important that it goes fast, then automation is encouraged. And that's why we also introduced the use of the ultrasonic bath to reduce the time for the sample pretreatment. We see that the interlaboratory results show that the reproducibility still ranges between 10 and 35%, and that there are differences in operational pretreatment procedures. Of course, the exchange of all this, our experience about the method is of importance. And that's why we started also to exchange this information. And it's also always in the framework to reduce the variability. Via our mirror committees with the reference lab, but also with our recognized laboratories, we exchange this experience and we hope to improve our variability. Thank you for listening.
Thank you very much, Chris, to give us a nice inside view about all the tricks and tips about clay determination by the standard ISO method. I think it's very good to see how laborious this method is, but also to see how the different steps can have an influence on the results, especially as we see that this RSD can be rather high. This is not the best thing we want to have. We want to have as low RSD as possible. But anyhow, we manage if we use some automation in some cases to get it lower. So maybe this will be the next step to go on. But up till now, I understand that's not that easy for all laboratories. So
Yep.
Thank you very much once more. So now we come to our first Q&A. There are already some questions in the Q&A. And I asked the speakers being Axel and Chris and maybe some others to join these Q&A sessions. First of all, I start with some questions for Axel. And I read the questions. I don't know if everybody sees the Q&A. I don't think so. But there was a first question about what do we mean when we talk of the soil carbon quality? So Axel.
Yeah, there are different possibilities to measure soil carbon quality. What we measure are the carbon fractions because they are really meaningful, and fractions mean the particulate organic carbon, the POM fraction, and the carbon that is associated to mineral surfaces, to clay minerals, for example, which we call the MAOM fraction. And if we know we have, for example, a high percentage of POM, and then the carbon is more labile and can be turned over faster as compared if we have mineral-associated stabilized carbon. And that's what we mean with carbon quality.
Okay. And a second question is about what is the major contribution to your clay depletion?
To be honest, it was difficult to find out what this question was about. So clay depletion, I tried to answer it. So clay depletion is really a long-term process that we are not looking at because this is really something in the soil development or if we have erosion. So therefore, maybe the question need to be written more precisely, or you can speak up and ask again, please.
Okay, so maybe you can, the person who put the question can please add some more information about his question, then we will come back to Axel. There's another question still in the same region of that. Which has the better quality, particulate organic carbon or mineral associated organic carbon?
We are again with this better or worse, degraded or healthy. This is really often beyond science, and it depends really on the function we want. So if we want to have soil organic carbon function to deliver nutrients, then we want to have a fast turnover, and then particulate organic carbon is better. If we want to have soil organic carbon to store carbon for climate mitigation, carbon sequestration, then we want to have it stabilized. then mineral-associated carbon is better because it's higher protected. So it depends really on the function you're looking at, and then we can judge on what is better and what is worse.
Okay, thank you. That were already some questions for you. Then we go over to Chris for some questions. And the first one is, and when I go a bit into the different steps of the sieving and pipetting method, the grinding to these 2 millimeters. What about when there are some particles with diameters above 2 millimeters, and if you start to grinding it, don't you think that these samples, their diameter can become less than 2 millimeter, and how does it affect, in fact, your methodology and your outcome for example, sand and clay and so on? So what about the grinding, and how does it affect the end results?
Yeah. The sieving at the level of 2 millimeter is a general applied method. Of course, if you have really tough particles that you cannot destroy with your fingers and to remove it less than 2 millimeter, then you will lose a part, but it's generally applied to do it at a less than 2 millimeter fraction. We also normally mention, if we see at our sample preparation that there is still a lot of sample left on our sieve, we mention that so that we also note it, that we are aware of that. But of course, it's a general applied method that you have at that phase. And of course, you have to try that most of the sample get through the sieve of 2 millimeter to get a proper result.
Okay, thank you. A question which comes back quite sometimes, what is the more exact methods, the hydrometer, Boyukov's method, or this method, this ISO standard? It's about all things about the procedure itself, the outcome, and so on. So do you have comparison between these two methods? What can you tell about it? It's quite some people ask about these differences because the hydrometer or Boyukov's method is also used because of some laboratories having limited resources. They also say we don't perform the treatments or the removals consistently. Can you correct for them? Can you correct the results afterwards for not having removed these things? So it's a bit about the whole methodology comparison between two methods.
With respect to the hydrometer, I think we can refer to the talk later of Maria. She will talk to that and give an idea about the comparison of it. Of course, every method has its specialty and always you can get the different results, but I think Maria can answer that question later on in her presentation. For the removal of the organic matter and so on, it depends also how much is available over there. If there is a lot of organic matter or carbonate presence, then it can have an influence on your clay sample, on your clay results. We can see if you don't do that, most of the time it is limited, but often if you don't remove them, you can have different in clay results up to 5 to 6 to 7%. So it is really an impact that you have to remove the organic matter and the carbonates to do a proper clay determination on the sample. So that is of importance.
Okay, thank you. And going together with this one, there was a question about adding farm manure. So feeding the soil, of course. Does it have any impact on the content, on the clay content of the soil? It doesn't have, I think, an impact on the clay content of the soil, but as Chris just mentioned, you have to remove it because it adds a lot of organic carbon, in fact, and in that way it can have a lot of influence if you don't have removed it. But the clay content itself, of course, does not change. As Axel also mentioned, clay is rather stable in the soil. The textural triangle being presented was the Belgian soil textural triangle. We know there are the USDA soil triangles with different classes and so on. You also have, you always have to use the textural triangle, which you think the best matches for you. So we have one in Belgium, so we use this one. But of course, we know of all the other ones too. There was then another question you mentioned a few times temperature of 70 degrees, but what happens if temperature goes above that 70 degrees? What can be the impact, or why is it important not to go above that 70 degrees?
I don't know the impact yet, because we did a test always at that temperature, and we kept it at the same range, especially to get a reproducible results. You have to define it at a certain limit, and also what is practically applicable also with an ultrasonic bath. If you go too high, it's difficult to obtain a bath at that temperature. But I don't know exactly what is the influence on with the higher temperature.
Okay, thank you. And there's also a question about fine and coarse silt. Can you use this method? And I think I can answer this method, this thing myself. Instead of pipetting twice to divide the clay and the silt, you can pipette twice or 3 times or 4 times, depending on whatever fractions you want. But then you have to recalculate the time you need before you take the sample. And I think this, unless Chris wants to add something, but I think this is the answer to that.
Yes, it's also included in the ISO method that you can do multiple pipetting, and then you can also get more differentiation in it.
An important one, I think, is about measuring soils with very high organic matter, organic carbon, peat soils, histo soils. Can they be measured with this method, or is the influence of that very high carbon too high to get nice results?
Yeah, normally you can perform the removal of the organic matter. If you do that properly, you can remove it, and then you can get a good dispersion. But then probably you will have to add more hydrogen peroxide to remove all the organic matter. You can see it on the intensity of the reaction of all your organic matter is removed, yes or no.
Okay, thank you. Maybe Bruno, you can add also. I think you don't measure if there's too much carbon in the soil, or
the big question is it still ecologically irrelevant by removing more than 50% carbon from a soil sample? What is left over? But normally you use so much hydrogen peroxide that it's almost not possible to remove everything in the pipette method for the sedimentation. So we don't do it if there is a lot of organic matter inside the soils.
Okay, thank you. There's also a question: have you seen comparisons to laser diffraction methods? We come
to that later.
We come to that in the other section. We come to that. We will answer that. We hope you will give you some nice insights into. There's one, maybe I don't know if you know the answer. Is the method affected by room temperature during measuring, as it is the case by using the hydrometer methods? Or is there no impact of the room temperature?
I don't know the hydrometer method in detail, so I cannot answer that.
Okay. Some answers to questions are already been given, but one important, I think, there was a question. The ISO method is amazingly misleading, and this is not the clay we know, somebody tells. My question is, have you compared it with the hydrometer method? And Maria, one of our next speakers, has given some publications on that. There are in the literature publication about soil grain analysis by the dynamometer method, comparison to pipette and hydrometer. So there are quite some references in literature. So please have a look if you want to have a better insight onto that. I will go on.
Can I answer? I see
the sure.
Now also about the room temperature. I misunderstood the question.
Oh, sorry.
Is this method affected by room temperature during measuring, as the case of the hydrometer method? It's important that you noted that the clay methods, with the pipette method. It has to be done at a fixed temperature between 20 and or 30 degrees Celsius. But it has to be done at a certain method because it's important, depending on which temperature is used, the pipetting time can differ if you work at a fixed insertion depth. So yes, the temperature is important to do at a fixed temperature.
Okay, thank you for this extra information. I think that way I went through, I think, almost all the questions. We are a bit behind schedule, of course, because we had one presenter who is unfortunately ill. So I propose that we now have the break for almost a quarter of an hour. And so we come back at 2:30 and then we will go on with the next session up till the end. So I hope to see you all at 2:30 for the following part of our webinar.
The host, should we start again?
Okay, very well.
Okay, let me stop sharing the break. Just want to encourage again panelists to have a look to the question and answers. If they want to have a look there, there are some more questions that were added during the break. Feel free to answer if you want. Thanks. Back to you.
And maybe we'll come back to these questions also in the next Q&A. One thing I want to remind everybody, and what I did read in the Q&A, was that at least one person is very happy with the puppet method. I enjoy that method much more than all the other methods. So that's very nice to hear. Even if it's a very laborious method, at least one man enjoys this method very much, but I think a lot of people enjoy it, although it's fairly laborious, but it's a nice method anyhow. This said and done, welcome back. I hope you enjoyed the coffee or another drink. And for the coming hour, we will talk about alternative methods. We had already about some links in the Q&A, alternative methods for the determination of the clay content of soils, and of course the comparability with the sieving and sedimentation method. As already said, that standard methodology is laborious, takes quite some time, and uses a lot of chemicals. But we know that other, often quicker methods are already in use. But the big question is, do they deliver the same results? And therefore, we'll have a closer look at 3 alternative methods. So first of all, we will have a look at the near-infrared methodology, and Clémence Mariage of the Université de Liège, of the Walloon region in Belgium, will introduce us into this method. Clémence is the quality manager of the soil lab of Jean Blou Agrobiotech, which is the scientific and technical reference for the routine labs in Wallonia. Clémence, you have the floor for 20 minutes.
Thank you. You normally see my screen and I put it in presentation mode so you can see it?
Yeah.
Okay, great. So thank you, Jos, for the introduction. So my name is Clémence Mariage and I work in Jean Blou Agrobiotech for a Recursive project. And so let me first explain you what is soil spectroscopy. So the idea is that you have a soil sample and you put a light source on it. And there are molecular vibrations and electronic transitions associated with soil constituents. And so the light is divided into 3 parts. A part is absorbed by the sample, another one is transmitted by the sample, and the last one is reflected by the sample. And the way the light is divided into these 3 parts depend on the constituents of the sample. And then you have a light receptor that captures the light that is reflected by the sample in a wavelength between 800 and 2500 nanometers. This is the range we work with. And then you can have the spectral signature of the sample, which give the absorbance in function of the wavelength. And this spectral signature obviously depends on the constituent of the sample. And so the idea is to have the spectra and then to convert it into a value of clay or total organic carbon, or we can also measure other parameters. But to do that, we need an interpretation model. So you need a spectral library, which is done by, which contain spectra and reference value that here in our case are done with the pipette method. And so the idea of this spectral library is to be representative of the values of the parameters, so a big range with very spread all over the range, so we have all the data. That is representative of the land uses, so we have here cultures, grasslands, forests, or vegetable gardens. and that is representative of the spectral diversity, so of the diversity of the soils. And we work mainly with composite surface sample in our case. And so we have 3 steps. The first one is a pre-treatment. So the idea is to clean a bit the spectra. And so to get a better view, get rid of the background noise, so clean it a bit. And then you can build a predictive model, so the link between the spectrum and the ref value. And to do that, you have different methods. You have PLS, you can work on global and local mode. And finally, you have the validation, like any other method. And when you do the validation, you have different indicators of performance. So you can have the determination coefficient between the ref values and the predicted values. The root mean square error of prediction that is huge, that is used a lot. Or the RPD, that is the ratio between the standard deviation of the ref values and the root mean square error of prediction. And so with these indicators, but you can see if the predictive model is good, or if you test several ones, then you can see which one is the best. And you can still improve the model, so improve these indicators by adding couples, so ref values and the near spectra in the spectral library. There are many advantages of using this method. So it's rapid and low cost. There nothing to see with what Chris explained, because it's just a few minutes to take the measure. It's reproducible, it's easy to proceed, it's non-destructive, so you can reuse the sample to make other analyses. It's environmentally friendly because you don't heat, you don't use chemicals, you don't even use water. And you can also predict several parameters with just one measure. So not only 3. And let's say, for example, we can predict the exchange capacity of soil, and with the ref method, it uses a lot of dangerous chemicals. And so here we are very clean. In Wallonia, so since for about 15 years now, we are working on the development of near analysis on dried and fresh soil. And so a general methodology has been developed. So we define an analytical protocol that is easy to proceed and repeatable. So all the labs of the network use the same analytical protocol. And we may test and finally choose best pre-treatments and predictive models for each analytical parameter. So not only for clay, but also for other ones, like first derivative, SNV, local or global mode, etc. So now in the lab, how do we do? So we use XDS instrument from FAO. Every day we make some tests. So there are performance tests that is some kind of internal measurements. And so the XDS gives us different values, and so we can check if it is okay or not. And we also scan a check cell. That is a cell that is completely sealed, completely closed. And so we don't need to fill in every day. So it's very repeatable. And we have to reach a defined value to check if the XDS is ready to work for the day. So the preparation of the sample for dried soil, we only make it dry. So air dried or in a stove at maximum 40 degrees, and then sieved at 2 millimeters, just like Chris explained previously. So it's really the same. And for fresh soil, we only sieved at 8 millimeters, and so it's measured really close to the sampling because it's fresh. And here we don't have any destruction of organic matter, of carbonates, etc. And so we don't use chemicals. We don't lose time with these steps. So we are ready to work after this first step. So of course we homogenize the sample, and then we fill in 2 quarter cups per sample for dried soils. and a control sample like you would do in the REF method, in any other lab, in any other method, and in any other parameter. And for fresh soil, we only fill in one ring cup, so it's not the same cups. And we also have a control sample. And so here I have a video of how we fill in the quarter cups. So for dried soils. So you see we put only like two coffee cups of coffee spoon of soil in the cup. And then we move it a bit to make sure that the whole window is covered by soil, because the light otherwise will reach air and not soil, and so the measurement won't be good. And so you see we have 2 quarter cups for each sample. And for fresh soil, we use a press. So we have here the place to put the ring cup and the fresh soil. And then there is a piston, and you need to push here with your hand to press. And so the piston come and push the soil in the cup, so that also the cup is completely covered. And then we can put the cups, so the quarter cup or the ring cup, in the FAO-GSP. And so it only takes like, I don't know, like 30 seconds to have the spectra afterwards. And then you can empty and clean up the cups with a vacuum cleaner, a tissue, or smooth brush, possibly with distilled water, but then you need to let it dry properly because water will influence the spectra. And so some attention points, like I said, the whole window must be covered by soil, otherwise you will measure air, and so you won't have a good measurement. The light of the spectrometer reach only a thin layer of soil after the window. And we recommend to work at room temperature because it's not always easy because not all the labs have a climatization, but at least at room temperature that is stable and the same temperature with between the samples and the instrument. So after all that, we have the spectra, and so we need to validate the measurement. So we have different steps for that. So for dried soil, as we have 2 measurements for the same sample with the 2 quarter cups, we calculate the root mean square. And so if this root mean square is under 10,000, then we say it's okay. We can calculate the mean of the 2 spectra. But if the RMS is above 10,000, then we need to rescan the sample. There might be an error somewhere, and so we need to take a new measurement. And for fresh soil, we only use 1 ring cup per sample, so we don't have that step of validation. Then you have the control sample, and so you can predict the clay content of the control sample and have a control chart like you would have with the REF method in your lab as usual. And so with this once the measurement is validated, then you can predict the clay content. And so you use the mean spectrum when you fill in 2 cups. You use one of the database depending on the land use, because in our case, we divided the database, so the spectral library, in 3 parts, so depending on the land use. So we have 3 spectral library. So you use then the defined pretreatment and prediction parameters. And we also use a repeatability file. It's a NIR file, so a file containing spectra of the same sample scanned in the different labs or the different apparatus of the network. And the aim of this file is to limit the differences between the labs. So if you go with the same sample in different labs, you should have the same spectra and so the same predictions. So this is the table of the kind of results you can have. So you have the clay content that is predicted here. And then you have 2 indicators that give you an indication of how reliable the clay you have here, the clay predicted is reliable. This is indicators that tell you how reliable these values here are. So the GH is the global age for mile and obese distance. And so when you make the prediction, actually, for example, you have like 10,000 samples in the spectral library, but you don't use all the spectra to make the prediction. So the software will choose like the 80 closest sample in the spectral library and use them to predict the new sample. And so the GH is the distance between the sample, so the new sample you want to predict, and the gravity center of these 80 closest samples. And the NH is the neighbor distance. And so this is the distance between the new sample, the one you want to predict, and the closest of these 80 closest samples. And so we define maximum values of these two indicators. And if we are above, we consider that the measurement is not correct. And so here in this example, all these values here in green are okay. So the measurement is validated. But here we have a sample with the NH that is too high. And so the measurement is not validated. Actually, the database or the spectral library doesn't contain samples similar enough to predict this new sample correctly. And so the sample will go in the other way, so go to be measured by the REV method. And so the idea then is to feed the spectral library and to put this sample, its spectra, and the REV value in the spectral library to feed it. So like I said, currently in Wallonia, we use the FAO-GSP instruments with an annual calibration verification and a database update. So the database and the spectral library, it's the same. And so this database is fed by the laboratories of Recursive Network. And so they are the most able to feed this library because they are spread all over the territory, so we can encounter different soils. And they work with the same reference methods and the same equipment. And so we are sure that the spectral library that is built is homogeneous and usable by every lab of the network, and especially with the same predictive model. We don't need to have different predictive models. And so here you have some values, some indicators of performance of how we work now in Wallonia currently. And so, like I said, with dry soil, we have two spectral libraries depending on the land use, but for fresh soil, we don't have enough data because we have less than 2,000 data in our spectral library. So it's not enough to spread it in depending on the land use. And here you have the ideal value that would be that is appreciable for these different indicators. And so you can see that here we can still improve the predictive models, but for cropland everything is fine. And here the root mean square error of prediction is some kind of approximation of the uncertainty of the method. And so you can see that here, especially for dried soil, for fresh soil it's better, but for dried soil we still have a big uncertainty of the measure. So there are still improvements needed and possible, but as it is a method based on the REF method, on the pipette method, we will never be able to be better than the pipette method. Thank you for your attention.
Thank you very much, Clémence, for your introduction to this near-infrared methodology. We see it gives already some options to measure clay fractions and other fractions, but some improvements are still possible. Having said that, we will have a look at the second alternative method, and that will be brought by Bruno de Vos. Bruno is a senior soil scientist at the Research Institute for Nature and Forest, the INBO in Flanders, Belgium. And what's special about him, well, special is maybe the wrong word, but he, almost 30 years ago, initiated the soil and analytical laboratory at that institute. So he introduced, in fact, the textural analysis into his institute. So Bruno, you have some 20 minutes to guide us through the laser diffraction method.
Thank you so much, Joost, for this nice introduction. Do you see the complete image of my presentation? Hello?
I think we have the different one because we see your notions and so on. Maybe you should take the other screen. Yeah. Bruno, your mic is muted.
So sorry. So this is the right presentation. You can see it. So I will start again. This presentation was made together with my colleagues from the lab, Els Menke, Anne Capieux, and Gerrit Genot. And we took the quite challenging title, Should Laser Diffraction Become the New Standard for Soil Particle Size Analysis? And the answer is yes, definitely yes. And I hope I can really show you some evidence why we should go to a new standard. Oh, that goes wrong. So laser diffraction technique. First, I can show you the outline. I will say something on the basics of the laser diffraction technique, then the procedure and accuracy. What are the relevant international standards that can be linked to laser diffraction? Is there any literature on laser diffraction and what does it say? And what is the comparison between laser diffraction and the standard method, pipette and sieve method? And also on the transfer functions and equivalent clay fraction limits that are used to link laser diffraction with pipette and sieve method, and then some take-home messages. So the basics of laser diffraction, the technique was already established in the 1970s. It's mainly based on a laser beam that hits soil particles. And what is very important, that is that small particles scatter light at wide angles and large particles scatter light at narrow angles, but with a higher intensity. So the main principle is that you have light, laser light coming in on a particle, and that some of the laser beams are diffracted on the particle. Some, if the particle is translucent, are absorbed, and there is some refraction. And we use often visible light because ultraviolet and infrared light is mainly absorbed. So the visual spectrum is used mostly in laser diffraction. So what you get by this angle is that you have a kind of scatter pattern like you can see here. And the intensity shows that here is the intensity for specific diameters, a higher intensity, and the angle, of course, is linked with the diameter of your particles. So if you have a system that sends a beam through a flow of particles, and all these particles are scattered, you have this scattered, then you can link with that detectors. You have different levels of detectors in the mid, low, and high angles. And on these detectors, you can detect the intensity, which is linked with the distribution of the particles. Okay, so all these theories, and there are two diffraction theories, Fraunhofer and the Mie model, assume that these particles are spherical particles, but we do know that most particles in soil do not fulfill that assumption. And this just leads to a broadening of the particle size distribution and usually higher mean particle diameters. What is important from these two models is that Fraunhofer model assumes complete diffraction, so no absorption of the particles, and the Mie model requires a homogeneous refractive index and also absorption coefficient but that is mineral specific, so it's really different for each soil mineral. What is the equipment we have at our institute? We started already in 1998 with the Coulter LS200, which is a device that could measure soil samples between 0.4 and 2000 micrometers, 126 detectors, no autosampler at that time. And the analysis time is between 19 and 90 seconds. And the cost of that apparatus at the time was 51,000 euro. While nowadays we use the Beckman Coulter. It's operational already since 2013, and it's equipped with a liquid module. Here, a bigger container of 1.25 liters, very important because we can sample the complete pre-treated sample, as I will show you in a minute. The same number of the channels and detectors, and about the same range we could measure, and that costed in the time 70,000 euros and 50,000 for the auto sampler that is linked to it. What is the procedure? We have start just like shown before for the pipette method, and basically it's the same method we are using for preparing the soil samples before they are analyzed with the laser diffraction system. We start from a fine earth sample, dried at 40 degrees. We take 1 to 5 grams of soil, 1 gram if it is silty, 5 grams if it is sandy, and if there is more organic material, we add some more material into the vessels here, into the beakers. We remove organic matter with hydrogen peroxide. We add demi water. We remove the water. We use baptization polyphosphates for dispersion, volume reduction, and then it goes to the autosampler. So basically, we use just the same pre-treatment as has been shown by Christine and others. What do we get as a result? This is a typical graphical output of the complete fingerprint of your soil from 0.4 micrometers to 2000 micrometers. So that's one of the benefits. We have a complete curve, a complete fingerprint of all the texture fractions. And we store in a standard in our database 2 micrometers, 50 and 2000 micrometers, but also what we call the equivalent diameters, 6 micrometers, 63. And I can show you later, for instance, we consider the clay fraction in the laser diffraction as being the fraction from 0 to 6 micrometers, silt from 6 to 63, and sand for 63 to 2000 micrometers. Here you can see that it is a sandy sample, and we also measure each sample 5 times. We have 5 replicates, so we can calculate the standard deviation for each bin, so for each detection. And so here you can see that, for instance, for the sand fraction, we have some uncertainty of what is measured in the sand fraction. These 2 bands here show some sand particles that are here, which means that we have a clear I have to move it. We have a clear fraction clay, silt, sand. We have a determination 4.1, 11.7, and 84.2% of clay in that sample, so we can classify according to the Belgian texture triangle and USDA texture triangle, and here it is a loamy sand according to USDA triangle. What comes out as output, and this is quite important, this is just a CSV list with all the bins. And we have, only for the clay fraction until 6 micrometers, 30 subfractions for clay. So we can, within the clay fraction, we can further see which are the dominant textures in each of these bins of diameters. So it's really much more in detail than just looking at the clay fraction, for instance, with pipette method. What is the relative standard deviation within the clay fraction? It's about 7%. And we also store the variability within each of these bins. So we can say that we have an excellent repeatability for each of these subfractions. I should mention that for sand and so on, we have upper RSDs saw almost up to 250%. So for some larger particles, it is not so accurate to measure them. Here you can see the control chart we developed. This is a control chart for 4 years. From September 2020 until now, the green dots are well within the 1 standard deviation of variation on our sample. Here you have 193 control samples. The mean is 17.3% of clay. We have a standard deviation of 1.5, which means on average a relative standard deviation of 8.4%. We used 3 internal reference materials for our control charts. So we have control chart. As you can see, there's only 1 outlier and then some alarming points, but mostly it is very nice in our control chart as we can measure. What kind of international standards do exist? Well, of course, the pre-treatment of soil samples, as has been presented before by others, and the normal standard ISO for sieving and sedimentation. But there is also an ISO specifically on laser diffraction methods, but not on soil quality. It's just saying, okay, it works on particles in 2-phase systems, so also including soil water suspensions, that's okay. Between 0.1 and 3 millimeters, okay. But it clearly mentions for non-spherical particles, as soil particles, the resulting particle size distribution is different from that obtained by methods like sedimentation and sieving. We don't know that, but what we really need now in soil science is an ISO standard for soil quality based on laser diffraction. If you look at literature, then it's amazing that you find a lot of papers already before 2000. People just looked at laser grain analysis and compared it with clay fraction, different models, and so on. And 20 years later, now, we still got these papers that just investigate the use of laser diffraction for soils. And also deal with how can we compare the results with the traditional sedimentation and pipette method. The big question is our soil science community not a bit too conservative? Because we do know that many labs already changed from pipette method because it's so laborious to the pipette, to the laser diffraction methods. Labs changed, but I think the soil science community did not really make that move until now. So what are the strengths of laser diffraction? It's a fast measurement, less than 1 minute, generates the entire particle size distribution. The order of magnitude is an order of magnitude more precise than conventional methods. It allows various sample pretreatment methods. In our lab, we use about 3,500 samples where we remove organic matter and carbon, but for other analysis, they don't remove organic matter to see in bentos, for instance, what's the impact. So you can decide with your laser diffraction what kind of pretreatment you use. It can be automated, and very important, it allows post-processing for factors such as diffraction models and optical parameters. After you did the analysis, you can apply other models to recalculate results. The weaknesses are that it requires a consistent and standardized sample preparation, that the amount of sample may be limited. Sometimes we use, if in some instruments they use less than 1 gram, then it becomes very tricky to take a representative subsample. You can have problems with the pump speed, with how to correct for obscuration, that one particle is not hiding the others. So the density of the particles in your suspension, that's a very tricky thing in laser diffraction. And then which scatter model should we use, Fraunhofer or Me model? Here is a comparison between the laser diffraction and pipettes. In terms of costs, they are very low for the pipette sieve method compared to laser diffraction. You have a larger cost in investment. In terms of labor intensiveness, sample preparation is the same if you use the same preparation method, of course, but in the analytical phase, we have very little labor in just analyzing the samples. And that makes, for instance, in our lab, if we use the manual pipette, we have about 6 soil samples a day, while we have 60 soil samples a day with laser diffraction. You have discrete fractions, while here you have the almost continuous fingerprint. Very important is that also conventional pipette calculates in mass percentage, while in laser diffraction volume percentage. Auto-sampling is possible. Operator error is high in pipette methods and rather low in diffraction. And the definition of the grain size in conventional pipette sieve method, you use in fact two different definitions of particle size for clay and silt versus sand, while you have one definition with laser diffraction. And accuracy, we consider the accuracy much higher with laser diffraction than with conventional pipette and sieve method. And to have some idea of the costs, we can now analyze texture at 81 euros for the pipette method, and it's only 7 euros without personal costs. So personal is really very impacting the cost of pipette method, while laser diffraction is a 1/3, 27 euros, and 12 euros personal costs. So that's a big difference in sampling. If we compare these methods, what is really the golden standard? In the classical soil science, pipette method was considered as the standard, but the big question is it the golden standard? And that's why new techniques like digital imaging comes in, where you really look at the particles, how smooth they are, what the shape is, what the dimensions are. And this very interesting paper of Bitelli et al., recently published in Geoderma, showed that if you use Blunt-Altman plots for comparing two measurements, that digital imaging and laser diffraction is very close to each other. While if you compare laser diffraction with pipette method, then you see a hell of a difference. It's all negative. There's a big difference between both. But if you compare pipette method with sedigraph and sieving, then it's also comparable. So here it is shown that we use two different ways of looking at soil texture, while if you consider digital imaging as the real diameter, then laser diffraction is far more close to the real diameter than pipette and sedigraph techniques. You can, of course, try to convert, because we have all these data in soil science linked with pipette methods. And then, of course, you can make transfer functions like Svensson et al. Did here. And here, in fact, you say, okay, my laser diffraction is linked with my sieving and pipette method. And you get quite good R squared. So 91% of the variation is explained. You can even make equations without intercepts. And what is quite important is that you also convert from the mass to volume percent, because this is incorporated in these transfer functions. But another and more advanced technique like we use now mostly is to look at these equivalent diameters like we did already in 2001, by just using the pipette method, sieving the clay sucking the clay fraction, putting it in a laser diffraction instrument, and then we could see what is the equivalent diameter for 2 micrometer with pipette with laser diffraction. We found 6 micrometer for a lot of soils in Flanders was the equivalent diameter. Bitelli et al. Using a Malvern masticizer and a ME model found 8 millimeters. And then in literature, you can find almost everything between 4 micrometers, 4.6, 5, 7, 9. So it clearly requires lab and device-specific determination of this equivalent diameter, and of course, a validation of this diameter. But this way you can convert laser diffraction to, let's say, equivalent diameters for your textural classification. What are the take-home messages? Of course, most of you are already at home, but please consider them. Laser diffraction looks very differently to soil particle sizes than conventional methods. So due to the underlying physical laws and assumptions, laser diffraction provides the total fingerprint of the soil particle size of the fine earth, allowing recalculation of all the fractions you need. Therefore, we recommend also to store the complete fingerprint in soil databases so that you can do also post-processing. The equivalent diameter of the upper limit of clay fraction shifts to larger diameters, like between 6 and 9 micrometers, but this is depending on the sample pretreatment, the device you are using, the diffraction model, and the working conditions. Soil textural analysis is in the end cheaper, faster, and more accurate than the sedimentation sieving combination method. It is well suited for high throughput soil textural analysis, especially useful for large soil surveys. There is increasing evidence in literature now that when based on digital imaging techniques, that clay and silt fractions measured by laser diffraction better approximate the real particle sizes than the conventional techniques. And therefore, we think that really particle size analysis with laser diffraction should have a place also in ISO standards, and there should be, let's say, a no-you soil quality ISO for particle size distribution with laser diffraction. Thank you so much for your attention.
Thank you very much, Bruno. I understand from your title it was a bit provocative, but you have tried to let us all say yes to this method, but maybe there are still some buts and howevers to this method also. But anyhow, it was a nice overview of this alternative method. And next, and the last alternative method we will look at today is brought to us by Maria Kreimeyer. Maria is working at AgroLab Agrar und Umwelt in Germany, and she started her career as an analyst, became the quality manager, and at this moment she is the site manager of the agriculture laboratory. And she will present us the third alternative method being the dynamometer method. So Maria, you also have the floor for some 20 minutes, please.
Good afternoon. I'm very happy to be here and have the opportunity to present the dynamometer method for determination of soil particle size distribution. How already said, I'm Maria Kreimeyer, a site manager of Agriculture Lab, which belongs to Agriculture Laboratory Group. We have more than 30 laboratories spread over the whole Europe, specialized in agriculture, environmental, water, and feed and food analytics. Just imagine you have more than 8,000 soil samples in your lab waiting for the particle size analysis with the pipette method. It would take ages to do that. Of course, you can try to increase your pipette method capacity, but it requires well-educated and experienced staff, which nowadays is not that simple to find. And of course, it is expensive. It is not a science fiction story. It is the reality of industry and commercial labs. And it was why it was our motivation to try to find the alternative method to the pipette method. We heard already today that the pipette method is seen as official reference method, is accurate but time-consuming and laborious. It has a long processing times which led to delays and high cost. So the aim was to develop a method that maintains the accuracy while reducing the processing time and cost. The methods which we considered was a hydrometer measurement, laser diffraction, and aerometer measurements. Now I would like to take you on a journey into the past. Everything started 10 years, almost 10 years before. Many grain size distribution samples in sediments from the Żelazny Most is the largest post-flotation waste reservoir in Europe, located in West Poland, needed to be done. It was not manageable with a pipette and even not with aerometer. It is why in 2014, University in Wrocław and Arcanum company developed an algorithm and the dynamometer method. The first device was built in 2015, and between 2015 and 2016, the test and scientific evaluation occurred. In 2017, the device was presented to the lab, also to AgroLab. The device looked like that. And we were convinced by the validation data and also by potential of this method and how simple it is. But was it suitable for the lab? At that moment, the answer was definitely not. But the method had so much potential that we decided to join the project and start to develop the automatic system which can measure many samples. In 2018, the device was built, and between 2018 and 2021, the method was optimized and validated. In 2021, the method was accredited, and we started with routine measurements. For now, we measure more than 30,000 of samples and more than 100,000 of fractions. This is also a great example for initiative cooperation between science and industry. The university developed the method and the algorithm with Professor Jarosław Kaszubkiewicz. The Arcanum company with Michał Staś gave all the technical solution, and AgroLab with myself will make it suitable for the labs. What is the principle of the method? Actually, the principle is very simple. The determination of density of suspension is based on the weight changes of the float which is submerged in it. The float is hanging on the thin line, which is connected with a piezoelectric dynamometer. The calculation for calculation is used Stokes equation, and all the data from the dynamometer, from the temperature sensor is transferred digitally to the algorithm, I can say, and the results calculated. Here is the whole process. The first sample pretreatment, the samples needs to be dry and sieve on 2 millimeter. And then you have free choice of the sample pretreatment. So it means you can do, of course, as in the pipette method, just with the removal of organic matter. We don't do it. So it means you can decide which kind of pretreatment you want to use. After this, 60 or 40 grams of the soil needs to be weighed, set with the water and dispersion agent, and shaking mixing. This step is important. You can shake overnight or mix with a strong mixing machine for 1 hour. After this, you place the cylinder on your device and start the measurement. The measurement is absolutely automatic, and this is really difficult to do something wrong here. The all data are transferred and calculated the multiple fractions. The characteristic of the method. The range of measurement 0.002 to 0.1 millimeter. You can do with or without organic matter removal. I will come back to this when I'm talking about validation. So it means you have a free choice of sample pretreatment. The measurement time is short. Example, 40 samples, 3 fractions, about 6 hours. So you can measure many fractions in one run, and everything is fully automated. And you can also have also flexible choice of the fractions. And also graphical presentation of results is possible. Here I prepared a short video so you can imagine better how it works. So the cylinder can be placed on the barcode scans to the autosampler on the software. The autosampler can have also different sizes. The device is mixing itself, measuring temperature itself, even washing the float and the mixer. The software is very user-friendly and show you the status of the measurements. So now I showed you the method, but now the question, how good is the method? So let's talk about the validation. We took long time for the validation, few years and a lot of samples. When I'm talking about a lot of samples, I'm not meaning 10 samples, 100 samples. I'm talking about thousands of samples. And there were different approaches. The first, it was a comparison with existing methods, focused on pipette method. The second, proficiency testing and accreditation according ISO 17025. Evaluation of statistical data like LOQ, measurement, authenticity, reproducibility. And also many scientific publications were published. Comparison with the pipette method. Here's one example in a paper from 2018 from the Wrocław University. You can see here the validation not only for clay, but also for other fractions. You can see the regression equation and correlation coefficient. We did the same or similar with AgroLab samples. I took this example just to show you the results are similar, but the approach is here totally different. Here you can see the pipette method, don't get confused by the names. And here you can see the nanometer method. Here we did remove the organic matter. Here we didn't. So you see the results are still good. But I would say this topic is good for next presentation on another presentation. Validation step. Next step is the proficiency testing, for example, by pair, measurement of a lot of PT samples. You can see the results here. Also, the method is accredited in our lab since 2021, and we are having quality control charts with a lot of thousands of samples. Some statistical data, reproducibility, we evaluated with 11%, measurable authenticity with 24, LOQ with 0.5%. Here you can see with which amount of data we are working. Summarizing. What are the main advantages of this method? The method is comparable with the pipette method. The consistency of results is given. There are no systematic differences. We have very good validation data. Everything is in digital form, and you can measure a few fractions in one run. The whole process is simple and user-friendly, what reduces the workflow. It reduces the time of analysis and is still cheaper than the laser diffraction. So if I would give the conclusion in one sentence, I would say this is a great alternative for the pipette method. One more reflection on this point. The success of this project showed how fruitful it is to stay open-minded and curious toward the innovation, and how important is for science and for industry to work together. Thank you for your attention.
Thank you very much, Maria. So now having heard 3 alternative methods, I think they all have their advances and disadvantages. Which one is the best? I don't tell it right now. It's up to you, I think, to make your own choice and see how far you can go with it. Maybe it's interesting now, because we have quite some time, to introduce now a supplementary Q&A session, because we have all these alternative methods together, so we can speak together with some questions and answers on it. So I take the opportunity to introduce an extra Q&A session. I think this is the best one before we go to the last part. Otherwise, it will take us too long before we answer the questions. I hope Axel is still here, because there was one question left from the very first topic. And it was about the type of clay ilites or kaolinites. Is it influencing the clay sock ratio? Axel? I don't hear or see Axel. Maybe I can answer myself. I don't, as we only measure the clay fraction, but not the type of clay. And we only do the measurements of clay. We can compare it to the sock ratio. I don't think it very much influences this ratio, but maybe if we should know the type of clay, but I'm not quite sure if this will influence quite a lot of it. So, but that's maybe the answer I can give right now, unless other people can give some more information on that. No. Okay, then we go on. We have had some alternative methods, and there's some questions about it, and that's the hydrometer method. And it's told that why is the hydrometer method neglected? And ISO 11277 being made the international standard when the hydrometer method uses no harmful reagents and less energy. And in the same sense, it's told that it is simple, easy, no computer, fast enough, and so on and so on. So can the three speakers reflect on that? What about the hydrometer readings? Anybody of you, Clemence or Bruno or Maria?
Yeah, I know that hydrometer was used in the past for the Belgian texture analysis. Many people think that it was pipette method that analyzed all the soil samples, but in the practice, it was really the chain hydrometer that was used for more than 80% of the soil samples to determine soil texture. And there was a calibration between that hydrometer and pipette method as a kind of validation, but because there were also so many samples that at that time in the 60s, hydrometer was considered the best alternative for pipette method. So yeah, it's maybe it was best that also somebody using the hydrometer was presenting here something. I don't know which labs are using it in Belgium still. I don't know.
Do the people from FITO or from other reference labs know if this is still used in the Belgian or the Luxembourg labs?
I'm not aware of anyone who's using that in Belgium, no.
So that's the reason why we didn't introduce it, the hydrometer method. I also don't know of any labs using it, why we didn't introduce it today, but we don't have all the definitive answers after this webinar, so maybe for another webinar we can see if other methods around the world are also in use and what their pros and cons. It's only one webinar about this specific aspect of soil analysis, but it's definitely not the last word about it concerning to me. Another thing or another question is, which of the soil methods you have presented would be most effective for gypsum and saline soils? Of course, in Belgium, we don't have a lot of these soils, and also not in Luxembourg, I think. But what should be the best or most effective method? Can we give an answer on that? Does it have to do with the preparation phase, and then it doesn't depend? Is it especially important that you do the washing very good, or whatever? Do you need other chemicals?
Yes, I guess that indeed the preparation should take into account how to remove gypsum, but then it's different from the determination method. Huh? So for laser diffraction, I don't think it will largely impact if you remove the gypsum, and even with saline conditions, there's no problem. I think it's also kind of dispersing. for the soil particles. So I think you will be able, with laser diffraction, determine the particle size.
Anyone else?
It's also similar, I think, with the pipette method. Also in the ISO method, it's described that together with the removal of the salts, you also wash out your gypsum and you work on that soil further on for the pipetting. But you have to do carefully check that the washing is good performed. I think it's the same as with the laser diffraction.
Anyone else who want to add something, or can we conclude with that? Okay, then I go to a next question. Also, one of the questioners asked, we compared laser diffraction analysis to pipette and typically obtained 2 to 4 times less clay with the laser diffraction method. And I read that this is quite common. And the question is how to link the laser diffraction results to the pipette method if we consider the pipette method to be the reference. Do we always have to have a reference sample for each soil environment that we analyze? And on top of that, there's another question. If laser diffraction becomes this standard method, how to deal with the results that are different for different laser diffraction devices? So it's both a question about difference between devices and difference between the pipette method. Maybe this is for Bruno to answer.
Yeah, maybe to start with the last one. Indeed, we see differences between the devices, but also within the devices, the methods they are using, the number of detectors, and so on. So the only solution is that you make really strong calibration for your equipment and use international reference materials and other materials for looking at the diameters and calibrate your own device for that. And the first question, yeah, that I showed, or by these transfer functions, you could transfer from this diameter you obtain with laser diffraction with the diameter obtained by pipette method, or use these equivalent diameters. And I have the feeling that in most recent literature, this approach of using equivalent diameters is really gaining field, is becoming more important. And that's something that also the lab has to calibrate for its own devices and equipment they have.
Okay, thank you. And then there's a question.
Maybe if I may add, it's important, I think, that we see it as pipette method gives some results of texture based on Stokes diameters, and we store differently in our soil databases the laser diffraction value. So we consider it as two different approaches of looking at soil texture. And these conversion methods are just to compare both, but we consider it as equivalent techniques to look at particle size distribution.
Okay, thank you. Then we come a bit back to the NIRS, and the first question is, what happens if you disaggregate soil aggregates before you perform NIRS? Does the method prediction then improves or not?
I don't really have the answer. I think we should test, but the spectrum is influenced by the soil particles, the soil constituents, and also how the soil has been prepared. So the granulometry, if we see it at another, not at 2 millimeters, but higher, then the spectrum will be influenced. So.
And maybe this goes hand in hand with another question about spectral library being publicly available or being compiled by the laboratory itself. I think therefore it's very important to have your own spectra. When you say that we don't disaggregate and so on, we need in fact own spectra. That's what you're telling us then.
Yeah, you need to have the same ref method than the spectral library to be sure that the prediction is good. And so, yeah, you need, if you want to use the NIR method, then you need to check if you have the same spectrometer, because only the spectrometer will influence how the measure is made, because there are different cells, there are different stuff in the spectrometer itself. And then you need to check that the ref method is the same.
Okay. There was also a question about the measurements or the measuring instrument, commercial versions. I think there are a lot of commercial instruments, so we don't have to mention here all the names. We have seen some names and some instruments which have been made. So we will. You have to look, I think, yourself or contact the people directly if you want some more information about the instruments they are using now in the different laboratories.
Yeah, there are many suppliers, so you can. It's big, really.
Okay. Having said that, and having most questions, I think, again, answered. I've seen that some people are already, or once again, ask for the presentations and so on. We will send them afterwards, so you will get all the presentations and the webinar. You can have a review on it. It's taken up by us. We know that, and we know that everybody wants to look once again into all this information when this is finished. So you will have all the information afterwards. So what have we done already this afternoon? We have talked already a lot about the clay content, but our last topic deals in fact with methods complementary to the determination of the content. And so we will talk about the clay itself. And maybe this can shed a different light on the clay determination itself. There was already a question about ylites and kaolinites, and well, we will talk about the clays itself. And for this last topic, we have a duo presentation of Nathalie Fagel and Simone Marx. First of all, Nathalie Fagel will talk about the identification of clay minerals by X-ray diffraction. Nathalie is a full professor at the Department of Geology at the University of Liège, Belgium, and she is responsible for the Laboratoire Argile, Géochimie et Environnement Sédimentaire. She has an expertise in the mineralogy and geochemistry of late Quaternary sedimentary archives. That having said, Simone will complement Nathalie's talk with some interpretations and applications of mineralogy results. Simone has first worked as an agriculture advisor before becoming head of the Department for Soil Analysis of the Administration of Technical Agriculture Service of the Luxemburg Ministry of Agriculture, Food and Viticulture. Nathalie and Simone, for this last session together, you have the floor for about 30 minutes, please.
Can you see the screen actually? Is it okay?
Yes, we can see it clearly. You can proceed.
Yes,
thanks.
yes, okay.
Okay, thanks for the introduction. So I propose to discuss a little bit about the method of identification of clay minerals. Here is the outline of the presentation. So first I will present a few information about the structure and the classification of clay minerals. Then I will introduce the main methodology to identify them, that is the X-ray diffraction. And after that, I will give you some examples. So first of all, clay minerals are actually very abundant in both soils, but also in sediments. And they are characterized by a quite small size, micrometric in range, and they are also characterized by a sheet structure. According to those two properties, they require a specific method of study, that is X-ray diffraction. The clay minerals represent a quite diverse mineral family, but they are all characterized by a similar basic structure that is present on this slide. So actually, all of the clay minerals are characterized by at least one layer of tetrahedral of silica and one octahedral layer of aluminum. And the combination of those 2 or 3 layers will be repeat over space. And if we take into account the thickness of the tetrahedral layers, the octahedral layer, and the space between the interlayer space, we define what we call the basal spacing, which is an important parameter to identify the clay. Now, the clay have been classified according to several parameters. 4 main parameters have been retained. The first parameter is actually the number of tetrahedral layer that are combined with one octahedral layer. The second parameter depends on the nature of the octahedral cation that define what we call deoctahedral or trioctahedral clay minerals. Then we have to take into account the layer charge, and finally, the type of material that is present in the interlayer space. To summarize the whole family of clay mineral, we can say that we have 2 main types of clay minerals. The first one is made by simple clay minerals, meaning that they are based on the repetition of the same kind of combination of at least 1 layer of silica and 1 layer of octahedral. That is the case, for instance, for kaolinite, which is characterized by a narrow basal spacing of 7 angstrom. Then we move to other group of minerals, and all those groups are characterized by a combination of 2 layers of silica with 1 layer of octahedral. Illite is characterized by a basal spacing of around 10 angstrom due to the presence of cation in the interlayer. Then if we move to smectite and vermiculite, we have exactly the same combination of octahedral and silica layer, but in that case, smectite and vermiculite are able to accommodate water molecule in the interlayer. And this is responsible for an expansion of the basal spacing up to 15 or 18 angstrom. Then we have another group of clay that are chlorides, and we have still the same basic combination of layer, but within the interlayer, we observe a layer of hydroxide. In addition to those simple clay minerals, we have actually in soil quite frequently what we call mixed layers. Those are made by a combination of 2 or 3 simple clay minerals. They may be randomly distributed or regularly distributed, and they are quite very common in soil. So for all those clay minerals, the identification of the nature of the clay is related to the thickness of the basal spacing, and this parameter may be obtained by X-ray diffraction. So to obtain X-ray diffraction spectra, we need to use an X-ray tube, and it's a quite simple system. So you have a high voltage that produce a beam of electron. The electron will hit a target, a metallic anode, that's maybe, for instance, in copper. And due to the interaction of the electron beam and the metallic elements, this produces an X-ray beam. And here is an example of the spectrum of copper anode. And among these beams, we will select one of the characteristic line characterized by a specific wavelength. For instance, the wavelength of the copper, which is at 1.54 angstrom. And this monochromatic beam will be transferred to the crystalline structure of the minerals. The primary beam will arrive on the mineral structure with a specific incident angle, which is shown here. And then the detector of the system will be located exactly at an angle of diffraction that is the same as the incident beam. So for X-ray diffraction, the geometry of the apparatus is very important to respect. And this is based on a physical law that was defined by Bragg. And actually, for any kind of angle position, diffraction angle, you may calculate using the diffraction, the Bragg law, you may calculate the basal spacing characteristic of the mineral, taking into account that you use a monochromatic beam characterized by one wavelength. Now, to measure X-ray diffraction of sample, there are two main preparations that are possible. First, you may analyze bulk powder. So the preparation of the sample is quite simple. You just dry the sample at 40 degrees Celsius. Then you grind the sample by hand and you press the powder manually to avoid any orientation of the particle, and then you submit this powder to the diffractometer. Here is an example of the spectra that you will get on a natural sample. As you can see, you have the intensity of the diffract beam that is measured by the detector. Then you have the different angles. The angle will vary over the analyzed time. And here you can see that for each angle, we are able to calculate, according to the Bragg law, a specific basal spacing. And those are characteristic of the different clay minerals. So such kind of spectra may be used as an identity card of clay minerals. But now, as we are working on natural clay sample, and especially soil sample, most of the time we have an association of different clay mineral in the soil, and therefore we may have a superposition of the different reflection by the different minerals that are present in the sample. And according to this, we need to compare several XRD patterns after specific treatment to check for the eventual superposition of the minerals. So there is a second mode of preparation of the sample, that is the preparation of what we call oriented mounts. And this analysis is made only on the fine, less than 2 micron fraction. So the bulk sample is suspended in water, then sieved at 63 micron to remove the sand fraction. Then we use different reagents to remove the carbonates, eventually the sulfates. We rinse the sample with water, and then finally, the solution less than 63 micron is we will extract the upper centimeter of this solution after a specific decantation time calculated according to the Stokes law. The drop of the liquid will be put on a glass slide, and it will be dry at room temperature until we obtain an oriented mount. So all the minerals that are present, all the clays that are present in this sample will be deposits along the same axis. So we have a specific orientation that reinforces the X-ray reply of the sample. This preparation will be analyzed after 3 treatments. The first one is just the air dry sample. Then the same sample will be solvate with ethylene glycol for 24 hours, and then analyzed by the XRD. And the third treatment, we put the sample for 2 hours in an oven at 500 degrees, and then we do again the analyze. So I will now give you a few examples from natural soil sample. And I will start with an outcrop from Burkina Faso. You have here a figure where you show a laterite profile, and we have analyzed the different samples from this outcrop. And we analyze in particular here the clay less than 2 micron fraction that is enriched in clay minerals. As you can see, the XRD of this sample is characterized by a few peaks of diffraction that occur at specific angle position, and each angle may be converted using the Bragg law into a basal spacing. Here you see that the main peak is observed with a basal spacing around 7 angstrom. We observe also another peak that is the half of the principal peak, and those two peaks correspond actually to the basal spacing that is characteristic of kaolinite group. And what also is interesting is you can see that with the different treatments, we observe a total collapse of the peak after eating. And this is due to the fact that kaolinite is quite sensitive to eating, and we have a deshydroxylation of the kaolinite due to eating. So this sample is dominated by kaolinite that is produced by a complete leaching of the cation in the hydrolyzing condition of Burkina Faso. Now we may also analyze the bulk sample of the same outcrop. And again, in the bulk sample, you observe more peak of diffraction due to the fact that we have all the different minerals that are not present in the clay size fraction. We still observe the dominance of the kaolinite here in red, but we also observe the presence of other minerals, for instance, quartz here, some mica or illites, and other oxide like goethite or hematite. So at this step, we just do a qualitative interpretation of the spectra, but we have to go further to try to quantify the abundance of the clay mineral and all minerals present in the sample. To do this, we use specific software that allow us to do some what we call Rietveld refinements. So here you have a spectra, the bulk spectra of the previous sample from Burkina Faso. The blue curve corresponds to the initial XRD diagram on the bulk sample. And then at the top of this, you have the red curve that is actually a reconstruction of the profile, taking into account the different minerals that were identified in the initial sample by the qualitative analysis. And then for each of those minerals, we will adjust the unit cell parameter. We will adjust if there is any preferred orientation due to the presence of specific cleavage of the mineral. And by doing this, we obtain progressively a reconstruct profile that is more and more close to the measure profile. Another example here, I just report the main clay formation that are present in Belgium. So you can see, for instance, in the southern part of Belgium, we have some clay minerals that are delivered by the weathering profile of Ordovician shales. For instance, in Jean Blou here, we have also the same kind of clay produced by the weathering profile of Devonian shale in Liban, in the southern part of Luxembourg. And then for the northern part of Belgium, we have the clay from Barry, and Barry corresponds to a marine environment. We have marine clay sand of Eocene in ages. Then we have what we call the bone clays that outcrops in Rumst, and they correspond to marine clay sands, Oligocene in age. And finally, the fifth sample analyzed here is correspond to the Beers clay that are tertiary sandy clays, and they correspond to more or less to an estuarine environment. So we did the analysis of the clay fraction, the less than 2 micron fraction by XRD. Here I just report the result after the solvatation by ethylene glycol, and the different color give you the different clay that have been analyzed. As you can see, the sample from Beers, from Barry, and from Rumst, they are all characterized by a peak that correspond to the position of the smectite-like minerals. That means that in those clay minerals, we have smectite-rich samples that are sensitive to ethylene glycol solvatation and that will expand by this treatment. No, for the other clay from Liban or from Jean Blou, you can see that we have more contribution from kaolinite here at 7 angstrom, and for illite at 10 angstrom. And for Jean Blou clay, we have also a small contribution of smectite, because you can see that the intensity of the peak is less important than for the other clay from the northern part of Belgium. So we have a clear distinction in the clay composition of those formations according to the origin of the clay. We analyze the clay less than 2 micron fraction by this XRD approach, but we have to take into account that it's only a fraction of the sample. So you have to take into account the abundance of the clay size fraction within the bulk minerals. And then you have to recalculate the contribution of the clay fraction in the bulk mineralogy to have a complete overview of the mineralogy of your sample. As a last example, I will show you some German clays that were collected in the Westerwald, in the western part of Germany. And here I report XRD diagram of those clay. You have the 3 treatments, so the natural air-dry treatment representing black. Then you have the solvated treatment representing blue, and the heat-and treatment representing red. As you can see again, we observe a peak that is quite strong at 10 angstrom. It corresponds to the illite, and it is not sensitive to any of the treatments. The peak doesn't change according to the 3 treatments. Then we observe a second dominant peak at 7 angstrom, and in that case, we observe that we have a partial collapse of the peak after heating, meaning that this peak is actually a combination of kaolinites, but it also contains a fraction of chlorides. Now, if we look at the 14 angstrom peak, we observe that there is also some change after heating. A part of the peak collapse during heating, and this is due to the fact that the chlorides are probably whether they are progressively transformed into vermiculite. And to be more confident about this interpretation, you can see that the glycal pattern show an expansion of some part of the peak, and we have here an expansion around 14, 15 angstrom that correspond to the fraction of the chloride that is sensitive to ethylene glycol. Now, if we report the XRD diagram under heated condition only, and we report the different samples collected from the same profile, from the bottom of the profile close to the Paleozoic bedrock to the upper part of the profile, you can observe on the XRD diagram, so we observe the illite peak that doesn't change over time, But then you have the peak around 14 angstrom that show a progressive decrease and a migration of the peak from a position of 14 angstrom to 12 angstrom. This reflects the progressive degradation of chloride into vermiculite. And this reflects the chemical weathering occurring in the profile that we may track using the XRD diagram on the clay fraction. And with this example, I thank you for your attention, and I'm free to reply to any of your questions. Thanks for your attention.
Thank you, Nathalie. And now I invite Simone to go on with the presentation.
Hello. From my side, can you see my presentation?
I think you have the wrong screen, Simone. We see your Teams.
Do you see my presentation now?
Well, we see the presentation, but also your notes. So maybe you should switch to another one.
I'll try it again.
Yeah, looks good.
So, good afternoon, and thank you for the floor. I will go on with a practical application of the comparison of what had been exposed previously by Nathalie Fagel on the mineralogical results of soils, and we'll compare that to sedimentary clay on two practical cases in Luxembourg. This is a collaboration of Lionel Laide, which is in charge of the soil lab in our department, and of Rüdiger Butz-Braun from the same called mineralogical laboratory in Germany. I will present you a case study on two soil profiles from the northern part of Luxembourg, located on the Devonian shales in the so-called region Aislake, which belongs to the Ardennes. Geologically, we are in the lower Devonian, with apparent material of shales coming from primary metamorphism. Soils have a loamy texture with variable proportions of coarse fragments deriving from the weathering of these shales. The description and the characterization of these soils present several challenges for us. First of all, depending on the pre-treatment of the samples, for example, crushing or not the sample in a mortar, these highly weathered and soft coarse fragments can end up either in the sand fraction or in the silt fraction, and are there at the origin of the variability in the silt and sand fraction. Secondly, the loamy texture of these soils coming out of the classical particle size distribution does not really match with the hydrological properties of these soils that we can experience in the field. For example, soils have a good structure and are well drained, and this leads to a lower risk erosion in reality than that is predicted theoretically by the erosion modeling equation. And finally, the question of the major pathogenous processes is yet not finally decided. So we tried in our investigations on describing and classification of these soils to associate analytical results from classical soil particle size distribution to information coming from mineralogical analysis in order to see if this could help us get some answers on the question I have just announced. Now let's zoom in the region where we opened 2 profiles for this investigation. We had a first profile on which was located on a quiet plateau with a very special facies in a small scattered location. And the second profile was located on a gentle and elongated slope, very typical for the region. And this was also the main soil type of the area. Here you have the pictures of these soil profiles. I start with the picture on with the right picture, the typical dominant soil in the region. We have a lateral view on the pit, and this lateral view is parallel with the slope. We can see the parent material of shales in the subsoil nearly vertically oriented. All the other horizons above has slipped over this parent material by solifluction, and coarse fragments took the orientation of the slope. Coarse fragments grow in proportion and size with growing depths. On the left side, we see a local exception of a stagnosoil with beautiful albeluvic tongues and stagnic properties. The classification that you can see here, according to VRB, the last version of VRB, is can be solved for the typical soil that you can see on your right. And for the left profile, we classified it as a stagnosoil. Just to give this complementary information, the classification had been done according to the results that will follow in the next slides. What I could add too is that apart from skeletic and endoleptic, these soils have typically also a high carbon content. We are about 3.1, 3.2 organic carbon in the topsoil because of the climatic cold and wet conditions of the Ardennes. As I said before, we combined analytical results coming from our lab, classical analytical results with mineralogy that had been done in the laboratory of Rüdiger Butz-Braun in Germany. And the pre-treatment of the samples are comparable to what had been explained previously by Nathalie Fagel. So we tried to associate both analytical results to see if this could help to answer some of the questions that were raised at the beginning of my intervention. Here are the results. We start, first of all, with the more typical soil of the region, the profile on the slope. Of course, the result from mineralogy and particle size distribution are not directly comparable because of the methodology, but they are here represented together in a histogram for a better comprehensive comparison. The left bars for each horizon are the results of the mineralogy, and the right bars are the ones from particle size distribution. We have 5 different horizons, AP1, AP2, which was a former plow horizon, then a B horizon going from till 45 centimeters, a BC horizon, and then the shale had been analyzed also, but only from the mineralogy point of view. From the mineralogical point now, we can see that the proportion of clay minerals and primary minerals, for instance, quartz, feldspath, and plagioclase, are nearly identical in the parent material, the shale, and the different horizons of the soil. So apparently there's no influence of alloctone aeolian material coming from far away. We also can observe in the pit that periglacial solifluction took place on the slope, but there is apparently no lithic discontinuity between parent material and solum. Of course, there is a high discrepancy between sedimentary clay and mineralogical clay. Mineralogical clay is more than twice higher compared to sedimentary clay, which brings me to the question, which clay determination would be the right one for the indicator sock clay that was proposed in the soil monitoring law? From the particle size distribution result, we know that reproducibility for the silt and sand fraction is low in this kind of soils because of the influence of the coarse fragments. But the sum of both fractions is quite stable in this profile. Together with the homogeneous mineralogy along the profile that we can see, we suppose that both fractions are mainly made up of the fragments of these coarse fragments with changing diameters of the altered shales. Furthermore, there is no clear evidence of clay illuviation in the B horizon. This brings us to what I have announced before, to a classical pedogenesis of a cambium soil. Now, if I switch to the details of the clay minerals, we see that chlorite and illite are the dominant clay minerals in the solum and in the shale. So clay minerals in solum are directly inherited from parent material. For further comprehension, it has to be said that illite and muscovite cannot be differentiated in XRD. They are both represented together in the green fraction of the bars here. As illite derives from the mineral muscovite by alteration, we would like to assume that this proportion is higher in the upper horizons. But sedimentary clay did not show significant increase in topsoil. So we conclude that the largest proportion of this illite muscovite fraction is made up of muscovite and belongs in the particle size distribution to the silt and sand fraction. And this could be part of the explanation of the differences between both methodologies, maybe also could be part of the explanation of the good infiltration of these soils. Now I come to the second profile. The second profile, we see a scattered, a small scattered curiosity of inherited albeluvic tongues from ice wedges and stagnic properties from periglacial time. Again, we see no lithic discontinuity between parent material and solum. But the parent material is different to the former profile. We have goethite appearing through the whole profile, a hint of former intense weathering processes. In the reductimorphic zones, so these are the clear zones of these alveoluvic tanks, goethite is absent because of iron leaching under the reducing conditions. As there is no clear evidence of clay increase in the surrounding oxymorphic zones, the qualifier of alveoluvic glossy in VRB cannot be applied for sure. If I come to the details of the clay minerals in detail show absence of chloride in the whole profile, except in the topsoil. We attribute this presence of chloride in this topsoil to the influence from the surroundings. The presence of kaolinite, on the other hand, in the whole solum is evidence of deep in situ weathering assumed coming from tertiary times. This would mean that this paleosoil, because of its topographically protected position against erosion and solifluction, had been impregnated later by perglacial processes altogether preserved till present times. And to conclude, some pictures that are assumed to deceive sand fraction after particle size distribution of the topsoil and of selected subsoils of both profiles. It confirms our hypothesis of contamination of the topsoil in the left profile, in the second, from the parent material around. Because if we look at the pictures on the right side, which represents the typical soil on the slope, and compare it to the picture of the AP horizon of the profile on the plateau, we see similar coarse fragments, and they have not the same aspect as the sea horizon of this profile located on the plateau. From my previous explanations, I conclude that for the soils developed on Devonian shales that present several challenges on the analytical side, and as well as on the description and classification side, because of low reproducibility of sand and silt fraction, because of the loomy texture that does not really match the property, especially the hydrological properties of in the field. The mineralogical determinations that we realized did not help directly and could not be compared directly to the results of particle size distribution, but at least it helped understanding the structure of the soils, and also to get some hints about past pedogenesis processes. They helped in the classification exercise, but what has to be clarified in future is, for instance, the question, what clay determination methodology in general are we going to use to apply the soil-clay ratio in the future soil monitoring law? And another question still important to understand, especially in agricultural domain, is the good structural stability and infiltration, how does it get an explanation? Thank you.
Thank you very much, Nathalie and Simone, for this two presentations. I think it shed another light on clay mineralogy than we usually think of. And with these presentations, we have finished all our presentations for today. Now we can still have a Q&A session. I've seen that a lot of questions have already been answered. So if there are still some questions, you can still put them in the Q&A. I will see if I forgot some important questions also about the first two parts of our webinar. Maybe one question I have missed, and that's one for Chris, I think. What about the distraction of the clay structure by hydrogen chloride acid? The United States Geological Service is using acetic acid instead. Do you have an idea about that and the influence?
I have no experience with acetic acid. We always use the hydrogen chloride to remove the carbonate content. It's also mentioned in the ISO standard that it should be added the hydrogen chloride content, but not that much, so that it's enough to remove it, but not to do it too much added. But with acetic acid, I don't have any experience.
Okay, thank you. There's a question about, maybe for Nathalie or for Simone, can we use mineralogy to explain the water retention capacity of soils and also the chemical fertility of soils? I see it was answered already, but maybe can do it orally too.
Yes, if I can reply. So it is clear that if you have a soil with swelling minerals like smectite or eventually vermiculite, those minerals will accommodate water during the wet season, and during the dry season, the water will be evaporated from the clay, so you will have an effect on the texture of the soil. So it's very important for the behavior of the soil. And I don't know what's next in the question.
Can we use mineralogy to explain water retention capacity of the soils?
Yeah, there is
and chemical fertility of it.
Actually, we to increase the efficiency of fertilizer, we use clay minerals. We add clay particle to soil, for instance, to increase the time action of the fertilizer, as far as I know. So it is clear that there is a relation between the presence of swelling minerals and the capacity of water retention of soil, but it will be very sensitive to season changes.
Okay, Simone, can you add on the chemical fertility of soils, or is that not of big importance?
Knowing that elite, for example, in my case study, is present means that clay minerals could release potassium, for example, and could be considered as potential stocks of potassium in these soils. And if we use extractions in fertilizer recommendation analyses for farmers, we also see that, for example, potassium or even magnesium, which is one of this cation stored in the interlayers of chloride, clay minerals, which are also present in the northern in these loamy soils presented. Both cations, like I said, potassium and magnesium, appear as present in sufficient at a sufficient level in the soil analysis done for fertilizer recommendation, and the consequence is that the recommendation for external in fertilization, especially mineral fertilization, is lower.
Okay, thank you. There's one big question in the chat to Nathalie also. After having the mineral composition in clay particles, should we consider the bulk mineralogy, or should we consider the percentage clay particles found by particle analysis in order to have the proportion of each mineral in the soil sample? So is it about the bulk mineralogy or about the percentage clay particles found by the particle analysis? Which one is the most important, or are both important?
I already replied partly to the question in the chat. So what I did, I tried to interpret the bulk mineralogy using some refinement analysis. So I'm able to give the abundance of both the non-clay minerals and the clay minerals all together. The problem is that the clay minerals are not only present in what we call the clay less than 2 micron fraction. You may have some clay minerals that are present in the silt fraction, for instance. So illite, chlorite, or even kaolinite may be fine silt rather than just present in what we call the clay less than 2 micron fraction. So there is always a confusion between clay fraction, that is the clay fraction in terms of grain size, and the clay mineral composition in terms of mineralogy. So it's quite tricky to reply exactly to the question. You have to take into account this confusion.
Okay, thank you. So I see some questions which goes beyond also our webinar. I think maybe they can be answered one by one, but I don't think they add to our webinar. I think we had most of the questions related to our topics. So having this said, and before giving back the floor to Filippo and Christophe for a final wrap-up and the closure of the webinar, I, from my side, already want to thank all the presenters for the very nice presentations, and all of you participants who joined from all around the world. I've seen from everywhere around, from Asia to Africa to the Americas. Thank you very much for your participation and your questions. And I hope that this webinar has shown you that clay in its broadest sense, and it's both the clay minerals as the clay determination, as the clay in relation to carbon, deserves our full attention, and that the last word on the determination and the use of it is not said. And now, Filippo and Christophe, for the last time, the floor is yours for the next 10 minutes. Thank you very much, everybody, from my side.
Thank you, host. I don't know if Christophe, you want to go first or?
Go ahead, Filippo.
Okay, so you can close the meeting. I will be very fast. I would like to thank again all the panelists, all the participants. Thanks again. Just to remind you that all the presentations and the video recording will be displayed on the BESOLAN webpage in the coming days. I would like to thank also Maria Romic, the chair of BESOLAN, for her kind participation. Well, we listened today that there are many methods available and many protocols within those methods. So we know traditional methods. Some of the new techniques have been also presented. So thanks again for sharing that. We hope that there will be a change also in that. There will be a dialogue between the different methods. Of course, taking into account the national context is the niche lab context is, of course, there are laboratories handling a huge amount of samples, laboratories with higher finance, larger financial resources, so they could implement new methodologies, while other laboratories have lower amount of samples, have limited resources, so they're still mostly relying on the traditional methods. I think this is very interesting, and the idea is that sharing the concept, the way the methods works is a first step to the change, let's say. But I mean, as a overall conclusion, I would say that clay is key. We hear today the importance also on clay for because it's very sensitive to change into management. It can be used as an indicator, and it's very important to take into account when we formulate fertilizer recommendation to farmers, especially for the highly reactive clays. So that is key. Measurement is key because we have to manage clay and properly, and but to make it accessible, as Axel was saying in the beginning, we need laboratories to work on that. We need laboratories to measure clay. We need protocols to support laboratories in measuring clay and making those measurements comparable and interpretable and, of course, reliable. This is why Glosulan plays a role in that. I want to announce that the next Glosulan Global Proficiency Test, also called ring test, also called interlaboratory comparison, different names for the same activity, will take place in the last part of this year, I guess in November, and we will include particle size distribution analysis in that. So will be one of the parameters we will include in exercise alongside carbon, nitrogen, phosphorus, pH, electrical conductivity, but particle size distribution is there. And actually that has been already included in several proficiency tests organized by region. Last one is the Asian laboratories that are grouped into SEALNet are about to launch a regional proficiency test, and indeed particle size distribution is there. They will use both pipette and hydrometer. So also the results of such exercise give us an idea of the comparability among the methods, something that was already discussed today. So definitely something that we are working on. Also in terms of harmonization of protocols, we are working on an SOP, a standard operating procedures for particle size distribution, taking into account both hydrometer and pipette method. Hopefully later on we can include other methodologies. So this is what we are doing. This is what Glosulan is doing on that. Definitely is key, and I will now give the floor back to Christoph, telling you that I really can't wait to hear about the next webinar organized by BESOLAN because these are so successful. So I really encourage you to organize another one. I encourage all the attendees, all the participants to reach us if they want to organize other webinars with international networks, from grouping together their colleagues or members of their laboratories. We can organize it together on any topic that is relevant for the network. So thanks again, and over to you, Christoph, and thanks again, host, for the nice moderation.
Okay, thank you, Filippo, for these nice words. So in name of BESOLAN, first would like to thank Joost indeed for sharing and moderating the questions. Very good job, thank you. Also you, Filippo, for always helping and hosting the webinar and keeping us on a short leash. That's very nice. I also want to thank all speakers for their excellent presentations, giving us a perspective on a very clear way and within time, and really widening our knowledge. I would like to thank all the participants as well for your attention and the questions that have been raised. So basically, to conclude, at some point I realized that the method of clay determination is probably older than any participant of the webinar of today. So, and as wisdom come with age, that's at least what they say. The webinar has shown us today indeed a glimpse of alternative and complementary methods that we are using in Belgium and Luxembourg. But indeed, I noticed as well a very large contribution of international participants. So I can only hope that it was a very inspiring for you indeed to enhance or to improve the determination of clay in your country in the future. And as Filippo said, there are a lot of things going on in BESOLAN. So I'm really also curious to have a look forward on that and to see how we can improve and make the results more comparable. So with that, once again, thank you all. And I want to conclude, and let's hope we can see each other next year on our next webinar. Thank you. Bye.
Bye.
Goodbye. Thank you very much.
Bye,
Goodbye.
thanks again.
Thank you. Excellent work.